My name is Veronica Ajewale. I'm an associate professor of pharmacy practice and the director for the Community Engagement Core of the NIH-funded Center for Biomedical and Minority Health Research here at Texas Southern University. I want to say a big thank you to you and the community members for joining us for today's program. We are really excited that we will be sharing a lot of great information alongside the experts of speakers and panelists that will be leading us into this conversation. So please have your questions ready. We would have an opportunity to take your questions after today's presentations. So thank you again for joining us, and we look forward to our continued collaboration. Thank you.
Good morning, everyone. Let's try this again. Good morning, everyone. Good morning. It's Research Day. Y'all have made it. Y'all have to clap for yourselves. Y'all went through 8 weeks of research. Good morning. My name is Sharmise Buck, and I am the graduate assistant assisting everyone on the P20 grant. And I have the pleasure of introducing Dr. Rain Rouse. And also, just some quick etiquette, cheer for each other, listen, cheer for each other, give everybody the same respect, and also, this is not a competition. Y'all are brothers and sisters, like y'all have built a relationship. So there's no competition, root for each other, all right? So, hold on, I just got to... Let me open this up. So Dr. Rouse is a pediatric cancer doctor and a physician scientist at Texas Children's Hospital and Baylor College of Medicine. She was born and raised in Houston, Texas, and enjoys nothing more than giving back to the same community that has given her so much over the years. She takes care of children with difficult to treat cancers and leads clinical trials testing new and less toxic ways to treat cancer. She is passionate about giving back and opening doors, especially for underrepresented populations, and leads community outreach and engagements at Baylor College of Medicine and within the Dan L. Duncan Cancer Center. In these roles, she focuses on ensuring all patients and families have access to the best treatments and creating a pathway for underrepresented students to pursue careers in STEM. She leads programs dedicated to providing research opportunities and mentorships to students locally and on a national level, and is so excited to be here today. So please welcome Dr. Rain Rouse.
Thank you so much, Sharmise, for that amazing introduction. We are so excited to be here today. I know that many of you, when you started this summer research program, had no idea what you were getting into. We just had a little pep talk in the back about how, regardless of age or stage, coming into a big academic medical center like Baylor College of Medicine and walking blindly into a research experience, whether that be in the lab, whether it be in the clinic, whether it be doing public health, can be really intimidating. Am I right? I have Dr. Catherine King here, who leads our MD PhD program at Baylor. Am I right, Catherine? Regardless of stage. And so we are so proud of you because you have truly excelled. I know that many of you in those first weeks and maybe even last week thought, I understand about 10% of this, maybe I understand 50%, maybe I understand 60%, and all of you are extremely high achieving individuals, which is why you were admitted into this program. The purpose of this program, this P20 Cured collaborative union between Baylor College of Medicine and Texas Southern University, is not to be limited to a summer experience. The purpose of this program is for us to watch you longitudinally grow, for us to be able to support you throughout that, whether that be in the form of career mentorship, whether it be in the form of us providing you an outlet for when times get a little bit tough in some of your academic studies, whether that be bouncing off of us what you're thinking about for your next steps. We really are excited to have you as a part of our family. And I said to all of you scholars this morning, all of our doors are open, our email boxes are open. We expect to hear from you, and we really want to keep you engaged in this program. We're really excited today because we have the opportunity to let you showcase your research. And I know that many of you have probably never presented academic research like this. I certainly had not at your stage, and I certainly had not in a very limited, short presentation. And so you all are learning and practicing a skill that many people do not even begin to think about until 5 to 10 years after where you are now. And so again, this is all friends and family here. We're all here to support you. Like I said to you earlier, there's nothing you could do up here on this podium that is going to make any of us less proud of you. You incoming freshmen, where y'all at, incoming freshmen? When you present your posters, again, we're all here to support you, and we're so excited. I'll also say that Baylor College of Medicine, this is a premier program for the college as a whole. Baylor is huge. This spans far beyond our Comprehensive Cancer Center. Our leadership is so proud of you. They know each one of your names because they received the brochure, and on behalf of Dr. Pavan Reddy, who wanted to be here but could not, and Dr. Helen Heslop, who was going to join on Zoom in a bit, I just want to welcome you. And they told me to tell all of you they are very much looking forward to you being a part of our Comprehensive Cancer Center in the future. I know Dr. Catherine King is looking forward to some of you maybe coming through the MD PhD program. And so we're just could not be prouder of you. I think I will turn it over to Dr. Ajewole because I know that she will talk a little bit about why this grant is so important, why it's so impactful, why it was the only one like it awarded in the entire country. Thank you, Dr. Ajewole.
Thank you very much, Rain. This is my sibling from another parent. I think we need to do a DNA test.
We're definitely related.
Yeah. Awesome. Thank you, Rain. Really great pleasure to be here. I'm very, very happy for each and every one of you. I echo every comment that Rain made. We are really, really proud of each of you. But before I go into my session, I would like to bring up the Dean of the college because of time constraint. Please join me in welcoming our Dean, Dean Mosavane, to share a few words with you guys. Thank you, Dean, for joining us.
Good morning. Thank you for the invitation. My colleagues at TSU, my colleagues at Baylor College of Medicine, thank you for doing this. Thank you all the students. I walked over here and actually looked at the posters. My background is I am a pharmacist and a pharmacologist, so I actually read and understood what it is you guys are doing. I'm super proud of what the students have done here. And if I could add one more item to what Dr. Rouse just talked about, it's a great experience in life when somebody throws you in the ocean and says, swim. It's intimidating, it's very frustrating, it's stressful, I have no idea what I'm doing. But down the road, that experience really pays off. And when it comes to learning, let me also share with you, there's two types of learning. There's a type of learning called passive learning, and you all know the passive learning. You sit in a classroom, I come in, I'm the professor, I click all the slides, and I read them for you, and you're just sitting there doing nothing, just listening, right? Then there is what you've done, which is active learning. You go to a lab and you actually do stuff with your own hands, so on and so forth, and that's the type of learning that really matters at the end of the day. So, welcome, enjoy your day. I'm again super proud of what has happened here, and I hope for those of you who are freshmen, let's not worry about the MD PhD program. Let's worry about College of Pharmacy. I want you to come to the PharmD program and then do residency in oncology, which is also a major residency, right? Dr. Ajewole will tell you more about that. So thank you for the invitation. Have a great day.
Thank you very much, Dean. I appreciate all the support that we've received from all the leaders here at Texas Southern University. So myself, I am a graduate of TSU. So graduated class of 2015, right? Go Tigers! Really, really excited to have the privilege again to be the PI and the director from TSU side on this really innovative and collaborative grant. If you all remember, during orientation, I said you guys are the firstborns, right? Because you had the inaugural class, of us doing this initiative. And what you're going to hear today over and over again is that you guys are truly trailblazers, because when you are doing something for the first time, you get to experience it live, and we make modifications, right? You learn, you experience, and you grow through that process. So thank you all for being part of this program for the past 8 weeks. I'm just going to go over some of the background information about the program itself, share some additional information before I open up the stage for you guys to, start sharing your presentations. So this is an NIH NCI funded grant, $2.4 million award, shared between Texas Southern University and Baylor College of Medicine. And I always pause when I see that red line that says the only grant funded in the United States this cycle. You guys should be proud and excited that you are part of this initiative. Really, really exciting. And not only that, this is the first time that we are having these grants in Texas Southern University. So imagine all the processes, everything that we have to, pull together to be able to have this program successfully implemented. The goal of the program is that we will develop a collaborative alliance between Baylor College of Medicine, a really huge academic center that you guys have experienced over the past 8 weeks, and for us here at TSU, focusing on cancer research, education, community outreach, training, and promotion of diverse biomedical research workforce, supporting a pipeline of collaborative, innovative cancer research pilot projects. And the faces that you see on this screen are the brain behind, the wheelhouse that wrote the grant and that are currently implementing the grant. Please help me to give an applaud to all of our team members. Every one of them, some are here in person, some of them are online on Zoom watching us, cheering us on as we implement the plan for this program. As you guys might be aware, this grant basically has 4 major pillars, 2 of them focusing on pilot projects. Some of you were directly involved in those pilot projects. Another pillar is the administrative core, right? For every grant or any research work to be successfully implemented, you need administrators. You need people, that think through the process, think through each of the steps. So we have an admin core. And of course, we have CREP, which is the Cancer Research Education Program. That is really the force behind this, the scholar program that you guys just completed over this past 8 weeks. And of course, in between that, we are very integrated. We have oversight. We have opportunity for program evaluation and making room for continued improvement as we go. As you guys can imagine, we need support, a lot of support from both institutions, and we want to appreciate the leaders across both institutions. You guys just heard from Dean Mosavin a few minutes ago, Dr. Monique Olaleye, VP Penn Marshall from Texas Southern University, even though both of them could not be here today because of other commitments and obligations, they both do send their regards, they send their appreciation, and they are cheering you guys on to great success. And like Rain mentioned, a lot of the leaders from Baylor College of Medicine as well, some of them not able to join, some of them are currently on Zoom. Also, within our program, we have internal advisory committee members, both from Texas Southern University and from Baylor College of Medicine. In addition to that structure, right? We also have another layer of structure called external advisory committee. So these are people outside of both institutions that are giving us directions, that are giving us guidance as we implement our program. So we have Dr. Upenda, we have Dr. Betty, Dr. McNeil, Dr. Ford, and Dr. Kuma. And you can see the names of their institutions all across the country, right? these people, they know that you guys are part of this program, and they highly recommend and they give us guidance as we implement the plan for our program. One thing I also want to highlight, for the audience, those that are online and in the room, is just looking at the structure of the CREP itself, knowing that the CREP is really integrating educational infrastructure. Over the past 8 weeks, you guys engaged in a lot of seminars, education sessions. You had opportunities to engage in research, as well as community engagement and outreach programs. And guess what? When we launched the application for this first cycle, we received over 70 applicants. I mean, that was overwhelming, like, okay, 70 applicants. But out of those 70 applicants, all 19 of you seated here came forth as the top, as the best. So you guys, we've really had the privilege to pour into you, to help you to shape on your desire for research and to be future scientists, future oncologists, pharmacists, or whatever else plans and goals that you have in your future. This, again, is highlighting what you guys have done over the past 8 weeks. And in those 8 weeks and beyond, like Rain mentioned, we are going to continue leveraging the community engagement core of both Texas Southern University and Baylor College of Medicine with our year-round engagement opportunities. So this is the calendar of all the major events that we have on TSU's side. And then in Baylor, they have a lot of opportunities too with the DLDCC community outreach opportunities. So we are basically here today to celebrate each of you for all that you've done over the past 8 weeks and looking into what you guys will continue to do in the year-round engagement opportunities. We're going to have presentations as well as poster presentations, from the rising freshmen doing poster presentation. The current postgrad or undergrad will be having podium presentations. Yes, Ray.
And I think we would be remiss if we did not point out that in addition to the funding we received from the NCI and the NIH, the institutional leadership from Baylor College of Medicine committed $100,000 a year to support you all. And the Dan L. Duncan Comprehensive Cancer Center has also supported funds, and TSU was very gracious in giving back the indirect funding, which is quite a bit. So this is just a testament to how important this program is, how important your futures are, and we're really excited about that.
Yes, thank you, Rain, for pointing that out. So thank you to the leaders of Baylor College of Medicine, Duncan Cancer Center, as well as CSU for all the support. The support is not just by mouth, but they also put their money, right? As they say, put your money where your mouth is. So money, financial support is something that is important, which we've received across both institutions. I'm going to skip this slide. The dean just shared his welcome or his greeting, and Rain was able to bring us greeting from the Baylor College of Medicine institutional leaders. Are we ready to begin the presentations? Okay, excited? One more. Oh, okay, I'm sorry. Yes, please. Thank you, Rain.
Thank you so much. It's just a real pleasure for me to be here today. I'm Catherine King. I'm the director of the MD PhD program at Baylor, and I have a lab, and I see patients at Baylor. I just want you all to know that we are super excited that you guys are part of this program. We need people like you to come join our field, to take care of patients, to do research that matters to patients. And you may feel like this is really intimidating, research is really hard, there's a lot of things that you don't understand, but I'm here to reassure you that that's how we all start. All of us here who have labs, who are working in this field, we started being intimidated by everything that happened. I remember when I was in college, it took me a day to read a paper, and I'd just be floundering through it. I'd go to lots of meetings. I didn't understand anything that was happening, but that's how you start. And we're really excited that you have joined this program. Y'all have done amazing work this summer, and keep in mind that we want you to come and join our field. So don't be intimidated to contact me in the future if you want help or guidance along the way, because we're excited that you're here. All right, excited to hear your science.
Thank you very much. Thank you, thank you. So we will begin now with our first round of poster presentation. It's my pleasure to bring up stage Dr. Murray, who will be moderating that session. Deborah Murray is a leader in equity, mentoring, and research education. And in 2021, she received the Norton Rose Fulbright Faculty Excellence Award in Educational Leadership. She was awarded the All of Us Evening with Genetic Research Program from NIH All of Us Research Program. Dr. Murray is the Director of Genetic Genomic Education Program in Human Genome Sequencing Center and an Associate Professor in Molecular and Human Genetic Department. She is the Co-Director of the Office of Community Engagement and Equity at Baylor College of Medicine. She provides faculty training opportunities and medical genetic training programs for M1, M2 students. She is a member of the Engagement, Communication, and Education team that provides community engagement, research for the Intellectual Development and Disability Research Center, and the Education Co-Director of NCI P20 Baylor College of Medicine, Texas Southern University Cured Program. Please join me in welcoming upstage Dr. Murray. Thank you.
Thank you. Good morning, everyone. This is an exciting day. We get to hear from you. Yay! I'm ready. You ready? All right. Cool. Cool. So just relax and take your time. Tell us what you know, okay? Stop studying and all of that. Just tell us what you know, okay? We're there. We are there. All right, so a few instructions. You have 6 minutes for your oral presentation. We have a clock back there, and so when it counts down to 5, I'll raise up my hand for your 1 minute. That means you wrap up, okay? And you stop at 6 minutes, okay? That's just the cutoff. And then we'll entertain questions. And I'm assuming questions will come from both Zoom and, okay, in the room. So we'll take a couple of questions and we'll move on to the next person. So your slides are here. You just click to find your slide. The next person should be right up, okay? All right, and I will introduce the first person. I'm just going to call your name so, you know, you can start coming now. But question about the clock. Is it going to just reset when they start? Okay, cool. All right, our first speaker is Nabras Abdul Rahman. And please repeat your name. Repeat your name if I said it wrong. Okay.
Okay. Good morning, everyone. My name is Nabras Abdurrahman. I'm a PhD student at Environmental Toxicology in College of Engineering. So I'm happy to present today. And my presentation is about to understanding and mitigating the cancer disparity in Texas, which is the comprehensive analysis of socioeconomic, healthcare, and environmental factors. So as you can see here in the introduction, there is why we are interesting to understand the disparities here in Texas, because if you can see, yeah, okay. Oh, so it's. So, because cancer is remain a leading cause of death in Texas with the significant disparity across this virus of population, and this is the statistic in 2023 annual report for case statistic. It was 140, almost 145 new cancer cases and 47,000 cancer death, and in children it's 1,370 new cases, is approximately 146 death. And from those, it's the most important, it was the lung cancer and the breast cancer, and here the statistic for both. And you can see the high rate of incident and the death of case cancer here in Texas. In lung cancer is 14,510, and the death it's 8,330, and in breast cancer 20,510 new cases, both female and male, and the cases of death it was 3,503. So before because here. Sorry, it shouldn't be in the result. This slide, it should be in the introduction. And here, also why it's important, because from those many research and many studies that it has been done, is showing many area in Texas, South Texas, Southeast Texas, or in Austin, or here in Houston, many cases and many cancer. Many cancer studies that it shows is still a very high rate of death here in Texas. So going back to hypothesis, that's why we hypothesize if we address our catchment is Texas, catchment area is Texas, and we want to address the region of disparties and why it has high rate of cancer death. So we hypothesize by addressing these disparities through their targeting the intervention and such improving the healthcare access, public health education, and implementing strength environmental regulation that will be reduced significantly the rate of death here in Texas. The methodology, actually, I worked with Dr. Rouse and her team, Dr. Ayala and Dr. Escobar, in two projects. One about the environmental factor, because how the environmental factor will improve. If we improve the environmental factor, it will be improved and reduce those rate of death in Texas. And the other project that I worked is about the healthcare accesses, and it was specifically most likely about the language barriers. And the methodology that we used, first we generate fact sheet with Cancer Alliance of Texas, and I become a member of the Cancer Alliance of member with Dr. Ayala, and I'm generating a fact sheet that with a project that they working now. And it will be this fact sheet. It will be produced, but it's still in the very beginning. And the first draft of the fact sheet that we generated here in the summer, and the other doing literature review, and the other project about the language barrier. We are working with health care. With public health care people and two doctors that give us access to children oncology, a group web, and we just right now we get it spreadsheet and full information. Then they will do analysis about the barrier and how they can improve that barriers to remove that barriers and make more people access to health care. So for the environmental factor, it's not included my result, the fact sheet, that it's separated in many tables. Sorry, we couldn't include that because they already programmed it and pull up the presentation. But here, the fact sheet that I generated, in the fact sheet with Dr. Ayala, and she was step with me. And we bought the global incident and mortality here in Texas, and it says that the statistic for that, the American Cancer Society, they expect it to be 136,000 new cancer cases and 42 cancer deaths in Texas, and that is very high, right? And then present what the sources of pollution. And the sources of pollution we did about many, like industrial emission and agricultural activities and vehicle emission, oil and gas. Then we analyzed what the key pollutants, and the root of these key pollutants, like air pollution, water pollution, soil pollution, all that it was created. And then we generate many environmental factor and cancer risk in Texas, and we put by regional, and we put many regions like in Houston, and what specifically kind of cancer that is affected the most, and the most environmental factor that I put here in the fact sheet, it was the air pollution, the molecular matter, 2.5, and ozone, and the industrial chemical, which is benzene, and we bought some from West Texas, which is also the red pollution, the air pollution, and the most important type of cancer, it was lung cancer, and the chemical is radon exposure, and in Austin, it was the pesticides exposure is the most affected chemical, and the most cancer, the most type of cancer, it was the lymphoma and non-Hodgkin and the prostate cancer. Then I put some initiated. What is the step? It should already the people have done. Some of initiative to reduce those cases of cancer. It was the Texas Cancer Plan and the Environmental Protection Agency and the Cancer Screening Prevention Program. All those programs, they work together, and they put too many things to reduce this cancer, but it's still this rate of cancer, it's increasing, and that's why we should identify some steps that we can do, like research and data collection, healthcare service, and make all that is need research, how we can facilitate it to people, to all people, and break this disparity or reduce disparity in a specific region. So, in conclusion, if we can address this disparity by research and innovation, community outreach, policy advocacy, and healthcare access. If we work hard for this point, for sure we can reduce all these cases and the rate death to minimum. And thank you. And I would like to acknowledge Dr. Rouse, my mentor, and Dr. Ayala, and Dr. Escobar, the lab member that I work with, and Dr. Shisha, Dr. Elujwa, and all the staff, Dr. Debra Murphy, Dr. Flora, and all the staff that they facilitate this program for us. Thank you.
Thank you. We'll take one question for Nabras.
The most death rate, it was in African American, then in Hispanic, non-Latino. And there is too many region, and the most interested region that we are interested now in Houston, the 5th ward, and Kashmir area garden, the cluster of cancer. There is many article, actually it says it's cancer clustering, but it's still no one have updated anything about those regions or what cancer, what pollutant that is cause the cancer. The EPA, I did look about that, the EPA and the city of Houston, they collect already sample, but they don't update anything about that. But so those, it will be interested area to know about it.
right. We also have a clicker so you can advance your slide, and you should be able to, trying to see, yeah. So a pointer as well, okay? So our next speaker is Nora. You need that. Right, yeah, the slide is, okay. And don't stand so far back.
Sure.
Okay.
Hello, everyone. Good morning. My name is Nora Abdurrahman. I'm a PhD candidate in environmental toxicology in Science College of Science of Engineering and Technology at Texas Southern University. I work right now. I'm working in my dissertation on emerging contaminants, and I was lucky to be matched in about the same area that I'm working on right now. So my topic for today is the impact of PFAS on liver cancer among subgroups in the United States, and it is an emerging public health challenge. So for the introduction, the liver cancer rates are getting higher in the United States, So here we just did for the subgroups, for Asians, for Hispanics, for African American, for non-Hispanics, white, and for Indian American. And with the years, it's getting higher, people diagnosed with liver cancer. So the hypothesis, it's exposure to pure and polyfluoroalkyl substance, PFAS, increase the risk of developing liver cancer in human due to their bioaccumulation in the body. So now you might be asking, like, what is the PFAS? PFAS is a group of human-made substance or chemicals, and they are also called forever chemicals because they bioaccumulate in water, in soil, and in air, and it's hard to get rid of them. That's if we can get rid of them. Before we go to the health impacts, here where they are found, how many of you prefer to use non-stick pans for their cooking? I assume most of us do, right? Lipsticks, the cosmetics stuff, cleaning stuff, or the waterproof coats that we use when it's raining, cleaning products, all of these has the PFAS products in it, and it's accumulating in our bodies. So what is the health impact of it? It's disruption of cellular process, also the mechanisms of, it's also the accumulation will cause immunotoxicity, neurotoxicity, and hepatotoxicity, which we are focusing on, which is the liver cancer that we are aiming to search for. So the method, so before going to this lab, they did a cohort study, and they actually did collect serum samples from 100 patients. 50 of the patients, they do have what is called cirrhosis, but they didn't develop it to liver cancer. And then the other 50, they have cirrhosis and they develop it to liver cancer. Then after that, after they have these samples, they did the PFAS extraction methodology to quantify it in the sample serum by using the liquid chromatography, tandem mass spectrometry, and then after that, right now we're in the stage, we're analyzing the level of PFAS in each of the controls and cases. Oops. Okay, so here's for the results. The first picture here, so it's the conversion of the PFAS levels in cases and controls. Sorry, I can go here, right? Okay, so for the cases, we see like all of them they have the PFAS, they are high in their PFAS, they are above the threshold, but in the cases, they do have higher rates of PFAS in their blood or in their samples. For here, we compare it, I try to compare it with EBA guidelines. And with the EBA guidelines, remember, both the control and the samples, or the cases, they have cirrhosis. So that's why it came out. So here, we have the threshold or the level of PFAS that we're limited to have in our bodies. But for both control and cases, the level of PFAS actually way higher than the recommendations. However, in the cases, it's much higher. It's exceeded the limitation that the EBA has in their guidelines. So in conclusion, or the key observation, the level of the PFAS or the BFOA, which is the peer fluorooctane acid, and the PFOS, these are both the most abundant PFAS, and I think during reading the literature, these two, they stopped using them in 2000. So they used them from the 40s, 1940, until 2000, until the EBA and other health advisory to stop using these two. So they are the most abundant. But for the level of these, they exceeded the limitation of EBA. That tells you 24 years later, they are still in their bodies. So you can imagine what we mean by forever chemicals. So what's a potential health, the health risk of PFAS compound associated with cancer, liver damage, developmental effects for the infants, immune system effects, thyroid disease, cholesterol levels. They spike the cholesterol levels as well. For the health, the public health implication, regulatory measures are crucial on the PFAS. For future research direction, further investigation should be done in that area, because still the link is not clear if the PFAS is actually developing the cirrhosis to liver cancer. And for the policy recommendation, we need more regulatory in putting these chemicals in our water, groundwater, in our food, in our cosmetics, and in our lifestyle as a whole. I'd like to acknowledge Texas Southern University and Baylor College of Medicine for this opportunity, and all the funds and all people who worked hard on this to make this happen for this day. Thank you so much. And I'd like also to thank my dissertation mentor, Dr. Bobby Wilson. He is not here, but he also made me, come and have this opportunity. So thank you. And also for the career development mentors, thank you, everyone. Thank you, Dr. Mary. Thank you, Dr. Rouse. Thank you, Mrs. Buck, Dr. Shisha. I'm sorry if I missed anyone. And it's time to have questions.
All right, we'll take one question.
These were serum studies. These were serum studies.
Excuse me?
The study.
Yes, they do have serum samples. Some of them, they were blood. Yes.
Was there a reason for using serum?
Could you repeat it?
The reason for serum.
The reason for serum?
Right.
To be quite honest, I don't know why they selected the serum of the samples. But again, BFAS get to our blood through the bioaccumulation in our body. And this is, I think this is the only way to measure the BFAS in our bodies. So, but I will definitely look for more information and get back to you why it was in the serum.
I have a question. What's the concentration of the BFAS that for the group that had cancer versus the group that didn't? Both have cirrhosis, but there is.
So the EPA has different, but when we do it, we actually compare the concentration of BFAS, the blood sample, then to the EPA after conversion, and then we count and compare between them. There is a biostatistician who works on this stuff and providing some of this stuff, but
Okay, all right. Well, thank you. Fascinating. Next, our next speaker is Taylor Boswell.
Thank you. Alrighty. Hi, everybody. My name is Taylor Allen Boswell. I'm a second-year graduate student here at TSU. In the COSET program, I'm studying biology. And I'm actually working on my research project right now and am due to give my oral thesis presentation at the end of this year. Okay, so circadian misalignment is correlated to many metabolic diseases based on epidemiological studies. So our circadian rhythm or our circadian clock is our 24-hour biological clock that assists us in helping keep our day-to-day activities in balance, such as eating, getting rest, doing exercise, and things like that. So when our circadian clock is misaligned, it can lead to many metabolic diseases, such as diabetes, mental health disorders, cardiovascular diseases. So what scientists use often when they're studying the physiology of the liver, they use primary hepatocytes. And what hepatocytes are the cells of the liver. They make up about 80% of the liver. So they use these hepatocytes as an ex vivo model of the liver physiology, ex vivo meaning outside of the body. And they use them as a tool in many scientific research fields. Certain types of hepatocytes that they use are hepatic stellate cells. And hepatic stellate cells are the fibrogenic cell type in the liver. And fibrosis arises and is present in almost all liver disorders involving hepatocellular damage. And the reason why scientists like to use, when they are studying the physiology of the liver, is because HSCs, they activate a variety of interactions with other hepatic cell types, such as macrophages, endothelial cells, cholangiocytes, which are cells of the bile duct, and also NK cells, which are natural killer cells that are part of the white blood cells in our immune system. So the main hypothesis in Dr. Guan's lab is to observe a connection between circadian remodeling and metabolic dysfunction associated with fatty liver disease and diet-induced obesity of mice. So some of the methods that they use to perform their research is genotyping. This is one of the first techniques that I did on my first day in the lab. And genotyping is basically the analysis of a DNA sequence of an organism to study the difference of the genetic makeup. RNA extraction, when you're extracting the RNA. Plasmid extraction, when you're extracting the plasmid from the DNA. SA-Cas9 plasmid cloning. And a big part of the activity that I did throughout the summer was mice liver perfusion. And what perfusion is it's used to cleanse the liver, and collagenase, which is a calcium-dependent enzyme, is also used to separate the hepatocytes from other cells so they can be cultivated. So cannulation is also a form that they use of the anesthetized mouse via the inferior vena cava. So in the picture here, this is obviously after we have anesthetized the mouse and dissected it. So you can see you can kind of see, it's kind of hard to tell, but we put it on the dissection plate, and we hook it up to the machine. I can't remember what the machine is called right now, but what we do is we cannulate the inferior vena cava, and what cannulation is we insert a tube or a needle in the blood vessel to collect the blood and wash out the liver and cleanse the liver. So it's a lot of things that go on with perfusion, so I'm really summarizing it a lot. But after we do the cannulation, what happens is the liver is excised, so we cut out the liver, and then we mince the liver on a plate with these long forceps, and the HSCs are then collected by removing the cell layer from the gradient surface. And then finally, we centrifuge to get the pellets to collect more the concentration of the HSCs. So the results the reason why we do this is to get a high yield isolation of primary mouse hepatocytes so that we can study the hepatocytes themselves. So all the isolations of HSCs from mice between 8 and 12 weeks of age typically yield 1 to 2 million HSCs per mouse. Mice older than 12 weeks or retired allow isolations of 2 to 3 million HSCs. So basically, the older the mouse, the more HSCs their liver will produce. And the vena cava cannulation increases reproducibility and requires less technical skills. I know in the original perfusion protocol, when they used rats, they actually would cannulate the portal vein instead of the vena cava, but they found that when you cannulate the portal vein, it doesn't reproduce as many cells as the vena cava, and it's more challenging, so they converted to using the vena cava in the rats instead of the portal vein. So the conclusion is basically to improve our understanding of the mutual control of metabolism and circadian rhythm to help us enhance our quality of life by promoting the circadian clock through the use of chronopharmacological and chrononutritional therapies. And the ultimate goal of improving the result of metabolic disorders is to identify the best times of day to rest, eat, exercise, and provide medication in order to restore the disrupted circadian machinery in shift workers and people with sleep disorders. And lastly, I want to acknowledge Dr. Rouse, Dr. Ajibola, and especially Dr. Shisia. I saw her every day on the 5th floor. And Ms. Deborah, Dr. Buck, Dr. Deborah, and Dr. Juan, And also my mentor, Dr. Guan, I don't know if he's on Zoom because he's not here, and also my bench mentor, Dr. Jiawa Wang. And that's it.
Questions? Well, while you're getting questions, are you, all of the techniques that you learned, are you using these in your dissertation research?
Yes.
Okay.
I forgot to add.
Is there, oh,
Does she have a mic?
Dr. Player?
Oh, okay.
Yes. Say that one more time. To validate getting the
Isolate the hepatocytes. When you isolate the hepatocytes, is there a way, like a marker you can use to validate that you're getting the cells?
How do you know there are hepatocytes?
How do you know they're hepatocytes? Because they look like hepatocytes?
I just, from my they were hepatocytes.
So scientific.
Yeah. They just told me that when we were taking them off.
Are there markers? I don't know the answer, but I'm curious. You're going to tell me later. Okay. Question?
Great job. I was just going to ask how this experience has impacted your preparation for your thesis and your kind of future goals, because you have more kind of bench experience than most people, and you got to do bench experience in a cancer research laboratory and use some of the same techniques.
Yes, it definitely helps.
Deborah, can you give her the mic, the microphone? Thank you.
Yes, a lot of the techniques that I didn't use in my current research project here, I used a lot of techniques I didn't know in Dr. Guan's lab, and it definitely did help me to prepare and bring other techniques to my research project. Definitely had to get used to the mice. I didn't know I was going to be working with mice, but I got used to them after a couple of weeks because we do have to go down and feed them twice a week because they're on like certain diets. They have to stay on certain diets. So yeah, that's pretty much how it helped me prepare. So I'll be using, I didn't know about like SK9, plasmid cloning and things like that. So I'll definitely be, have that up here when I'm working on my research.
Oh, thanks. All right, our next speaker is Devora Asham.
Thank you. Okay, do I just click this?
Yeah, that'll forward.
Okay. Hi, everyone. My name is Devora Ashkadal. I'm a third-year pharmacy student here at Texas Southern. And this summer, I've spent doing pharmacological literature review on medications used in cancer in clinical trials with Dr. Foster and Dr. McEvoy. So a little background, I decided to pick these two medications, which basically are used in treatment of osteocarcinoma cancer. And osteocarcinoma cancer is a kind of bone cancer that basically starts when the cells are forming the bone. It basically causes unusual and weak bone growth. And these are in relapse patients, so we want to see if these are safe enough to give to these patients. So my two drugs are ifosfamide and etoposide, and this was a phase 2 relapse patient. So a little background, relapse in cancer patient presents significant therapeutic challenges. Ifosfamide and etoposide are chemotherapeutic agents used in combination for various malignancy. This study investigates the efficacy and safety of these drugs in osteocarcinoma relapse patients. And we are assessing response rates and overall survival outcomes and importance of finding effective treatments for relapsed cancer patients. So the hypothesis is that the combination of ifosfamide and etoposide will show significant efficacy in treating phase 2 relapsed patients, improving response rates and overall survival. These are the methods that we used. So it was a phase 2 clinical study, so that means it has already went over the phase 1 study, which means that these medications were safe enough to use in relapsed patients. So the clinical study, the phase 2 has 27 relapsed cancer patient. Ifosfamide was combination medication that was given. Ifosfamide was 3 grams per meter squared, so we would have to get their heights to dose them. And this was administered on day 1 through 4. And etoposide was 75 milligrams per meter squared, which was administered on day 1 through 4 as well. And these were categorized patient response, complete response, partial response, minor response, and stable disease. And the overall survival was calculated at 12 months. So the results were 22%, which were 6 patients out of the 27 that achieved a complete response, 26%, which were 7 patients that had partial response, and 11%, which were 3 patients that exhibited a minor response, and 6 other patients also maintained stable disease. Overall, the survival rate at 22 months was approximately 30%. In conclusion, a combination of ifosfamide and etoposide demonstrated a notable efficacy. Significant portion of patients achieved complete or partial responses. Overall survival rate at 12 months suggested a promising therapeutic option. Further studies with larger sample size and longer follow-up are needed, and potential to optimize treatment protocols for relapsed cancer patients, specifically in osteocarcinoma. And here are my acknowledgements. Just want to thank Dr. Ajawale and Dr. Rouse, also Dr. Shi, Dr. Estes, and Dr. Muri for all their help throughout this program. And I also want to give acknowledgement to my lab mentor, which was Dr. Jennifer Foster. Thank you.
Questions?
Very good.
Thank you.
Very good, Devorah. Question, can you explain to us or define what does partial and minor response look like? How are you defining that?
The patients with osteocarcinoma relapse, they have these signs and symptoms, which were swelling, they're not able to walk, all these symptoms. So in minor response, so on day 1, they would have a decrease in those symptoms, but not like a complete response in these medication. Or first time, it wouldn't work at all. Like they wouldn't get the complete response that the other patients that had a full rate. I think she had another question.
Good morning. First of all, let me say excellent presentation. So my question is, was your clinical studies based on a particular type of cancer, being that relapse differs in those cancers?
So I had multiple studies. So I was doing pharmacological literature review, basically trying to see if these drugs would be good enough to go through these phases. I specifically decided to focus in osteocarcinoma relapse because there was a good survival rate, and there was, like I said, the follow-ups were most prominent with this patient, because there was an issue where patients don't follow up, so we don't usually get like the results that we want. And so I decided to choose specifically the osteocarcinoma, but I did work on a lot of others like solid tumors and breast cancer as well. Yes. No problem. Thank you. Do you like?
Okay, it's on now. Great job, Devorah.
Thank you.
I don't know if you said this in the middle of your presentation or not. Did you say how the drugs were administered?
I know the patients would because these were pediatric patients, so they were intravenously administered, and obviously we would have to get their height, but yes, it was intravenously, and I believe they had to come to the hospital so we had to monitor them throughout the day, the 4 days that they were staying, just to see how they were responding to the medications.
Okay, were there any outliers with the patients? Like, did some of them have an allergic reaction and you had to stop treatment immediately, or you didn't look at it?
No, this had the most promising result from the other studies that I was doing.
Okay, thank you.
Thank you.
Our next speaker is Ira Baggett.
All right, so my name is Ira Baggett. I'm a second-year pharmacy student at Texas Southern University. And so my study is assessing paclitaxel efficacy in AC711 breast cancer cells, a comparative IC50 measurements using MTT colorimetric assay and IncuCyte live cell analysis. And so breast cancer is obviously a leading cause of cancer-related mortality for women worldwide. And one of these reasons is the recurrence of breast cancer and metastasis represent major challenges. And so the cell line that I used was AC711. It's a triple negative breast cancer cell line, so which means that it doesn't have the receptors that are usually associated with a lot of cancer types, so it doesn't respond to hormonal treatments, which makes it harder to treat. So usually we use cell lines for in vivo studies, because obviously when we're testing these drugs, we can't use them in humans. And so when you can't use these hormonal treatments, one of the second line treatments would be chemotherapy treatments. And so paclitaxel is a chemotherapy drug. It's called a taxane. And it works by inhibiting some of the microtubule dynamics in the cell. And so we use this drug, and so basically we want to determine the IC50 of this drug in the AC711 cell line. And so basically my hypothesis was, so I used two methods, two assays. So I used the MTT assay and the IncuCyte. The IncuCyte is basically a camera that takes periodic images of the cells. So I wanted to determine the IC50 values, so I thought they were going to be the same. So for the methods, obviously we cultured the cells, so I'm not going to go through that. That's just a routine thing. So basically for the experimental conditions, the cells were treated with a serodilution of the paclitaxel at a concentration of 100 micromolar to 1 nanomolar. So it was a serodilution, so it was treated for 48 hours. I had a control group, which is untreated cells, and there was also a blank. So there was nothing in that well. So as far as for the cell viability assay, we did the MTT assay, so that was just per manufacturer protocol, so that's very routine. We use those assorbance values to determine cell viability. The IncuCyte, like I said, it just takes pictures of the cells growing. For data analysis, the cell viability, so we use the absorbance to calculate, we use the percentage of the absorbance of the treated samples relative to control. And for the IC50 determination, we use a dose response curve generated from the data from the cell viability calculations. And so I just wanted to go a little bit more in depth for the MTT assay. So basically, you see the cells on the plate, you add the MTT reagent, so you remove the media that was in there, and then you add the MTT reagent. You incubate it for 3 to 4 hours. And so during this time, the MTT will be reduced to formazine, I'm not sure. And that's what creates that purple color. And so then you add DMSO as a solubilizer, and then you'll get this color change that you see in number 5. And so on the far end of the plate, the dark purple will be the cells that are more metabolically active, so there's live cells, and then on the other end, you have the dead cells. And for the results, so basically, Figure 1 is the MTT at 48 hours, Figure 2 is the IncuCyte, and then Figure 3 is the images from the actual IncuCyte. So going back to Figure 1, on the y-axis you have the cell viability, and then the x-axis is the actual concentration of the drug. So both of my replicates, replicate 1 is the red, replicate 2 is the black, so that's the same for figure 2. And so you can see that, let's see, my IC50s were relatively the same for both replicates. But however, for the InQ site, you can see that there's kind of a wide variance between that. Some of that could be because of operator error. So actually, replicate 1, that was the replicate that I did, and then replicate 2 is my lab mentor. So she's a postdoc, so she has a lot of experience doing these kind of experiments. So basically, oh, hold on. So for row A on the top, so that's the highest concentration of the drug, and then at the bottom H, you have the control. So you can see that the drug has, it's killing the cells, it's effective. Columns 1 through 6 is replicate 1, and then 7 through 12 is replicate 2. So for the conclusion, obviously the InQ site is great for monitoring the cell morphology and proliferation, but however, things like the seeding density or your level of technique can affect the degree, the quality of your result. And so the MTT assay is like a well-established method. That's just one of the reasons why they use it. So it's based on the metabolic activity. So, the seeding density of your cells won't affect your results as much. And also, like the MTT is it's more easy to reproduce that result. So someone with not a lot of experiments, you can still come up with about the same result. So the MTT is more sensitive, but the InQ site does have some advantages because you can see the cell morphology of the cells. So there might be some way the drug is affecting the cell as far as morphology, so you can see that as well. So overall, it's best to combine both of these assays to get your result. And I think as far as for the InQ site, it could have been better if it really comes down to technique. So I think the IC values could have been a little bit more similar, but overall, the idea is to incorporate both of these assays to get your result. And for future studies, so basically we're trying to establish the IC50 and the parental line. So the next step would be to create a paclitaxel resistant line and establish the IC50 in that line. And that is that. And for my acknowledgements, yes. For my acknowledgements, I want to thank Dr. Ajawale, Dr. Rouse, and everyone who made this possible. Thank you so much. This has been such a great experience. Thank you. Okay. All right. Questions? Yes, ma'am.
Hey, Ara, that's a great presentation and well-designed study. Thank you.
Thank you.
I have a question about breast cancer in general. You said this is triple-negative breast cancer, and so chemotherapy is routinely used as a first line. What are other types of breast cancer? How are you usually treated? How are they usually treated?
So like I was saying for this, so you said triple negative?
Yeah, that's what you said, right? This cell line is a triple-negative cell line.
So, I mean, usually if they respond to like hormonal treatments, you can use things like aromatase inhibitors or like estrogen drugs, like estrogen inhibiting drugs. So like enzostrol, tamoxifen, those kinds of drugs. But being as they don't have the receptor, they won't really respond to this. And then you have like androgen deprivation therapy for like prostate cancer. So those are some ways they treat.
Great, thank you. Thank you, great job.
Okay.
Any more questions? All right, so we are coming up to a break. So first we want to thank all of our brave speakers who started the morning out. They did a wonderful job. Yay. And we're looking forward to the rest. We have 10 minutes and we do, 5, I'm sorry. Woo, they corrected me real fast. So you have 5 minutes, so you take a bio break. I think the restroom's over here, snacks, and just get up and stretch and move, and we will return.
Thank you all. As we go on break for the scholars, there is a QR code on the screen and also in the agenda. Feel free to scan that and complete the post-program survey. So that's going to be open all through the rest of the day. Thank you. And the snack table is also open. Please feel free to get some. Thank you.
All right, everyone, break time is over. All right, so we are getting ready to start the second round of presentations. And I actually have the pleasure of introducing Dr. Shea. She is also my PI. I am a part of her lab, so I get all the love and the tough love and get to learn from her. But Dr. Shea is a phenomenal professor of pharmaceutics at Texas Southern University. She has over 20 years of experience in preclinical drug development. She serves as the founding director of the Institute of Drug Discovery and Development at Texas Southern University, which is an NCI-designated subcontractor to provide service for investigators across the United States. She is the PI of the RCMI Center for Biomedical and Minority Health Research and the co-director of the GCC Center for Comprehensive PK/PD and Formulation, funded by the Cancer Prevention and Research Institution of Texas. She is also the program director of the graduate program in pharmaceutical science at Texas Southern. Whoop, whoop. Dr. Shea has 4 years of industry experience, has successfully obtained grants for over $30 million, completed over 200 collaborative research projects/services, taught over 1,000 PharmD students, and personally mentored 10 research scientists and postdoc fellows. Please welcome Dr. Shea.
Thank you, Chomsy, for the wonderful introduction. So we're gonna move forward to the second part of today's presentation. Can you, it's right here. So, Alex Bozar. She's one of my students. All right, coming.
All right. Okay. All right, good morning. My name is Alexiana Bosire, and I had the opportunity to work under Dr. Shea Wang, but her last name is Dr. Gao. So, introduction. Immunity does affect cancer, so it can either increase cancer growth or it can inhibit cancer growth. So when you talked about immunology in my lab when it comes to genotyping, we used different types of methods, and we focused on genotyping protocol in research. So the hypothesis that we were working on in Dr. Gao's lab was how the knockout mice will affect DNA strands in the final gel electrophoresis. So what we did do in my lab is we obtained mice from the Jackson laboratory, and we also had the wild-type mice that we did get from a local farmer who the lab gets mice from. So what we did in this experiment is we took the different types of strains of mice and extracted the DNA from both the control group and from Jackson, which had the wild type. And the mice from Jackson were called the RAG1 mice because they lack the B and T cells that helps in immunity. So in DNA extraction, in genotyping, we used DNA extraction, PCR, and gel electrophoresis. So what we had to do is pretty much euthanize the mice, both from the control group and from the Jackson laboratory. And what we did was put the samples inside the gel, whereby we analyzed the gel sample. As you can see here, so the first 5 samples that I had in my experiment, the first 5 are from the Jackson lab. These mice over here did not contain the B cells and the T cells, while here were the wild type mice that did contain the B and T cells. So if you look at the fragmentation, the ones that of the wild type had more DNA fragments as compared to the wild type. So what you see here is the ladder while we were comparing the wild type versus the RAG1 mice. So we used the ladder to compare the presence of the DNA fragments. So the expected results, according to JAX, was supposed to look like this. So they had more samples than I did, but you could see the empty wells right here. they did not contain the RAG1, the B and T cells. So in conclusion, if you look at my results, you can see that the RAG1 mice that did not contain the wild type or the B and T cells, we could go ahead and inject the different mice strain that different cancer strains that we had in the lab, so we could continue to do cancer research on how immunity does affect cancer. So in my acknowledgement, I would like to thank the P20 Cured team and my lab mentor, Dr. Shay Gao and Holly Kwang, because Holly Kwang was the one who was able to work with me throughout this whole process. Thank you. Any questions?
So I do have a question. You, in your hypothesis or method, you have several list method, then your result only showed the gel. So is anything else in the PCR DNA extraction? Is any, you can just talk about it.
Yes. Okay, when it comes to genotyping, so the 3 steps are involved. So the DNA extraction is whereby we had to go ahead and euthanize the mice, extract the DNA from that. So in the PCR stage, we had to take different samples and mix it with a green cocktail that had different types of proteins. So afterwards is when we used gel electrophoresis. So before we did the gel electrophoresis, those 2 steps were the vital ones. So the final step is what we needed to make sure that we go ahead and do the experiment or continue with our studies in terms of immunology in relation to cancer.
Is there any, Chomsky?
Okay, I have one question. So with your gel, was
Yes.
it agarose or SDS page?
We used agarose.
Okay, that's all I wanted to know. Make
All
sure
right.
you clarify.
Yes.
So people will know.
Yes, we use agarose instead.
All right, thank you. Thanks, Alex. Give us a good presentation about basics of biology lab. Let's move on to Taylor Farrell.
Hello, everyone. I'm Taylor Farrell, and I'm a senior in undergrad at TSU. And this summer, I had the opportunity to work in the Musto lab, also the Bentov Research Center. And the main focus of my research was identifying the structure of 7sk RNA in an in vitro model of MIRA9 triple-negative breast cancer. So triple-negative breast cancer is an aggressive form of breast cancer with a high mortality rate due to the lack of appropriate treatment regimens. And MYC is a transcription factor known to be overexpressed in most cancer, and it interacts with PTFB, which is a positive transcription elongation factor. 7sk RNA is RNA that plays a critical role in regulating transcription by isolating PTFB. So as you can see in this chart, a low MYC count means that the cell is normal, not precancerous, not cancerous, but as it increases and is overexpressed, it becomes more precancerous or cancerous. So our hypothesis is that the difference in gene expression leading to cancer development is due to MYC interacting with PTFB and changing the structure of 7sk to allow for more global transcription. So as you can see in the chart below, there's 3 different structures of 7sk. What we're trying to do is find which structure is apparent when MYC is overexpressed. So a method we used was DMS chemical probing, which is to identify the structure of 7sk in the MYC overexpression cell model, which is the MIRA9 triple-negative breast cancer cell. And what DMS is dimethyl sulfate, which is a chemical that probes the structure of RNA by adding a methyl group to single-stranded RNA. And the cell line that we used was Mira9 because it's inducible and it was a verified cell line from the Westbrook lab. And as you can see in the charts below, this is the methods we followed. So first we started with the modification, and then we went on to the reverse transcription to amplify the cells, and then library prep, which is where we see how intact the RNA is to the cell line. So we have an unmodified and a modified, so without the DMS and with the DMS. And then we have the sequencing, which we do, and we count the mutational rate. And then we have structural modeling, which we did not reach in this lab or this particular experiment. So as a result, we found that the cell line differences in DMS tolerance prevented us from producing libraries with the Mira9. And we were able to sequence the 7SK in the human cells, which was the HEC293 T cells, but further troubleshooting is needed with the Mira9. And as you can see in the diagram, we have the RNA, as you can see those bands. Then we have the HEC, which is the human, and the mouse, which is the 3T3, and then Mira9, which is the triple negative. And so, as you can see at the RIN scores, that determines how intact the RNA is to the cell. So it decreases with the HEC and 3T3, which it's supposed to do, however, not with the Mira9, and we think it's because of the amount of time that we left it in the DMS. We should keep it in there longer because it's a stickier and tougher cell. And so in conclusion, there were some limitations. RNA is easily degraded, and the cell lines, some of the cell lines that we used have little RNA, so it's harder to get good reads. However, the samples obtained were good enough to make libraries from the human cells, and the next step would be titration or changing the amount of time the Mira9 stays in the DMS to detach from the cell. And I would like to thank TSU and Baylor College of Medicine for giving me this opportunity, and also the CREP leaders and Bentob Research Center and the Musto Lab. Thank you. Any questions? Yes.
How many times did you lose your sample to degradation before you
Let me tell you,
figured it out?
For about 3 weeks straight, we kept losing it and kept losing it and kept losing it until we finally got a good sample. But when we got to the sequencing, that's where it was like, there's barely any mutation in it. So we had to keep trying and keep trying and keep trying until about recently. But I wasn't able to do the structural modeling part. My mentor has to finish that area. So I'll let you know if we ever find the model or the structure of 7SK.
Yeah, good job.
Thank you.
Great job. Great job. Looks like you had good positive and negative controls over that. So
yes.
What are the precautions to prevent degradation of the RNA when you extract it?
Okay, so we have to put it under the hood to keep like excess, germs, temperature outside the hood. So we had to put it in the incubator as well. And sometimes with the DMS, it's so strong because of the pH of it that sometimes it's out of our control. So, and even with the media, we used a different media multiple times, which was the MEBM and then the PBS, but it worked with the PBS but not the MEBM.
Great job. So wear gloves all the time?
Yeah.
Yes.
Any other questions?
Thank you, Taylor. That was a very comprehensive project. Okay, let's welcome to the next presenter, Chinizara Lajezi, if I didn't say it correctly.
Good morning. My name is Chinaza Jay-Z. I'm an uprising junior at Texas Southern University. My major in biomedical science. I was opportunity to work under the lab of Epidemiology and Popular Science under the supervisor of Dr. Oluyomi. My topic is all about the geospatial distribution of brain cancer, specifically in brain cancer because I'm more interested in brain. To be a neurosurgeon, I wanted to know more about it in that field. My introduction is like the brain cancer has like the rate of 12 rank in dead on brain cancers, and also the incident rate has the about of a higher level of concentration of brain death in higher regions such as America and Europe, and the lower regions such as in Africa. The overall statistics of the brain cancers in the United States has, in 2021, has approximately 1,777,566 new cases, and in 2022 it has like 608,366 people died. In Texas specifically has the amount of level of brain cancer death compared to Cuba, which has like every year has a reported case of brain cancer, about 2.5% of death. My hypothesis of brain cancer is the incidence rate exceeding significantly closer in central geographical area in Texas. The previous research on brain cancer, that was specifically for brain cancer in children from 2000 to 2017, where they were able to find out the factors that causes the brain cancer in children was based on social and environmental factors like petroleum environment, and they were able to find out that 54.3.1 million of children with brain cancer tumor in Texas. So in my method of study, I basically didn't really use specifically in children, but I had to put all age and all ethnicities to see the factors of how the brain cancer are really affected in our populations. And the study indicated the age adjustment incident rate in high and low of brain cancer incident in Texas by counties. The potential environmental and health factors influence in brain cancers we're able to bring out the data from the Texas registered cancer, and it was reported that 4,408 premium brain cancer and 500, compared to the first one, compared to the children report, the children report has a higher level of brain cancer report compared to the whole overall from children and adult age. So it has like a 574,295,892 population still at risk, and also 2,308,884 cases are still reported. In my result, I was able to differentiate the adult rates in age-adjusting incidence rates, both in children and all age. We're able to find out, like miraculously, that the study of brain cancer has been decreasing over a year from 2000 to 2021. So in my study, I was able to use the geospatial map. It's kind of like a more level of coding, where I was able to create my own map, which states that from 2000 to 2021, we're able to notice that the first map is the basics of the by counties of brain cancer reported, and the second one is from 2016, like 16 years gap. We were able to notice that it really increased in areas by county, and from 2022, it kind of like decreased. In our own factors, we're able to know that because of during that period of time, COVID-19 kind of like came and no one were able to report the statistics of brain cancer reduced or increased. And from 2020 to 2021, it went back up, which is for our future researchers that are going to know that why it really increased in some counties. in my conclusion, we were able to analyze the environmental influence on brain cancer and the potential impact on public health strategies and identifying the risk factors. This is from the whole map from 2000 to 2021. Shows by county the adjustment incident rate of brain cancer in Texas. I'm so thankful for this wonderful program that was able to push me out and experience more things in all that environmental. As a future surgeons, I come to realize that we are more advanced in technologies, and it's very important to have the skills to understand that it's not about being there, working with patients, but also be able to know the statistics and the causes of those diseases and know how best to work with our physicians. Thank you. I'm going to acknowledge this wonderful program with Dr. Adjegule for this opportunity. This is my first time, and Dr. Rouse with her mentorship and words really pushed me up. And for the TSU mentors, thank you so much.
Thank you, Tyler, for that wonderful presentation. I do have one question. It's this one, right? So can you explain this slide a little bit more? I saw the line has been pretty flat, but according to your next slides, it's actually have increased over the years, right? So what does that mean?
This might indicate like the level of the concentration of brain cancer in Texas. Concentration of brain cancer in Texas from 2000 and 2001. From 0 to 10 shows the incident adjusting rate, the age adjusting incident rate in both from children to all adults. The green one indicates the adult rate, which categorically increased from 8 and is decreasing from the year 2000 to 2001, we can say that it's decreasing, but the children is kind of like going up from 2000 to 2021. And for all age, mixed out both together, is going through a level of decreasing, but let's say a little bit of the adult decrease compared to the children. The y-axis is the age adjusting incident rate. These are, oh, sorry, all age. Just my, no, it's, okay, sorry. The 2000 to 8 for the adult shows the level of period that the adult had was reported of brain cancers in Texas by county.
So I was just a little bit confused about the y-axis they have. If it's age, the age up to 10, right? So how the adults can be under 10? That was my question. But maybe your y-axis have different meaning than I interpreted. Sure. All right, thank you, Taylor. All right, the next one is Elijah King. All right.
Hello, my name is Elijah King, and I'm currently giving a presentation on, I'm a second-year pharmacy student here at Texas Southern University. I'm currently giving a presentation on tenacin C mitigating antibody effects on pediatric high-grade glioma growth in vitro. So first of all, what is pediatric high-grade glioma growth? So what is it? It is a aggressive and highly lethal tumor growth that has a median overall survival rate of 9 through 11 months. In the image to the left, we can see the picture of the tumor itself, of what it looks like in the thalamic region of the brain. And then here on the overall survival with patients with pediatric high-grade glioma, we can see at the year 2 mark, it is a less than a 25% chance. So we can see how deadly it is. So then the current treatment strategies, now, of course, the main thing that you're probably thinking of, why not just remove the tumor out by surgery? Well, the way the tumor is set up, rather than being one large mass, it is actually broken up into smaller masses and diffused throughout the brain structures. So therefore, the surgical resection, therefore, is not possible. So the main treatment lines of therapy are chemo and radiation therapy. So here, the alternative treatments that are currently under study is the chimeric antigen receptor, or CAR T cells. Now, these CAR T cells are engineered T cells that are not only made up of human components, but also components found in nature. So, and then it's also been shown throughout study that they slightly improve survival, but they encounter challenges in the clinic. And for the purposes of this presentation, I want to only focus on one of those challenges, which is the immunosuppressive tumor microenvironment of the glioma. So on this immunosuppression, so the main thing that causes this immunosuppression of tenascin is by a protein called tenascin C. And tenascin C is a extracellular matrix protein that is expressed during development, wound healing, and is also not only expressed in those areas, but also expressed in, of course, the glioma cells themselves. And here on the bottom left image, we can see how it is expressed in, of course, all of our high-grade glioma cell lines. So for the purposes, only focus on the orange and the yellow bars. And then here I have a graphic in the middle detailing what the mechanism of action is. So what it does, how it binds and stabilizes transforming growth factor beta, leading to decreased T cell proliferation. And of course, that is a problem for our CAR T cells, because of course, decreased T cell proliferation, the CAR T cells are not going to be able to go in and of course do their effective job, which is in neutralizing the glioma cancer. So how can this immunosuppression be overcome? So two ways. We have a neutralizing antibody and we have CRISPR-Cas9 mediated knockout. So I'll only be reviewing, of course, the neutralizing antibody, which is the first way. So which leads to my hypothesis of mitigating tenascin C with monoclonal antibodies will decrease growth of pediatric high-grade glioma cells in vitro. So the methods that we did, it was that we conducted a 5.2-fold titration or dilution of tenascin C antibodies. Those two antibodies, 2138 and 3358, those are the same antibodies. However, what they do is they bind to different positions on the tenascin C protein. So we basically set it up like that, and then we incubated it for 7 days, and then we quantified it using bioluminescence. So here are the results. So what we did was we normalized our results to a PBS control, so phosphate buffered saline. So of course, when we inoculate those cells with phosphate buffered saline, of course, it's just saline. It's not going to do anything. So the cells are therefore all supposed to be alive and accounted for. So after we normalized it, we also did an isotype control, which is an unspecified antibody. That binds. It's just an unspecified rat antibody isolated from a rat. And then we could see throughout our results, throughout the differing concentrations, there's relatively no change of the cancer growth, which leads to my conclusion that tenascin C mitigation with monoclonal antibodies does not affect pediatric high-grade glioma growth. And then, so the future directions that we could take this, we can culture the cells next time with both antibodies simultaneously. You know, we have two proteins binding to do two different positions at the same time. Maybe it's going to produce a higher effect, maybe not. And these next two, knocking out the gene, the next two bullet points on the bottom, these are currently ongoing efforts that my mentor, Jack Tremblay, is currently working on. You know, and then later, hopefully with preferred results, we can test the cytotoxicity of the CAR T cells against the knockout tenascin C cell lines. And then one of the strengths of the experiment is that the controls that were used strengthened the validity of the experiment. So we had the PBS control, of course, and the isotype control. But the main weakness is that the bioluminescence does not give us a direct cell count. And I would like to thank the P20 and TSU program for sponsoring this program. And I would like to give special thanks to Dr. Jamie Anastas and Jack Tremblay, who gave me guidance, of course, on the wet lab work and proper data presentation. And here are my references.
Great job, Elijah. It was a very hard topic. I'm glad you make it easy for us to understand. Deborah?
I have two questions for you. First of all, what other type of technique could you have done for the cell count? And give us your hypothesis on what the outcome of the experiment they're working on with the knockout. What do you think is going to happen based on some of the data you're seeing?
Okay. I'm not familiar with the other processes associated with giving us a direct cell count, so on that one, I can't really give you an answer on that one, but I can answer your second question, which was sorry about that. Can you repeat that? Oh, the hypothesis of the next experiment. Okay, I got it. Sorry about that. So the hypothesis of next experiment, I do believe that, of course, when we're testing the CAR T cells against the knockout tenascin C cell lines, with the tenascin C cell lines, when they're knocked out, of course, they're not going to be able to express the tenascin C, therefore decreasing its immunosuppressive ability. So with the CAR T cells being able to, they will be able to effectively do their job, which is, of course, to neutralize the glioma cancer cell growth. So I believe that we will be looking at great results. I feel like it will effectively neutralize the cancer cell growth.
Sounds reasonable, but you're going to look up the other one and give me an answer later, right?
I do. I will.
Hey, Elijah, great presentation. The pathway is very clear. Reminded me of my postdoc study. I was doing the TGF-beta pathway as well, actually BMP, and I knocked out the SMAD1. So I really like the rationale and the pathway presentation you made, and also the future direction. So you can see that even that those two antibodies did not have differences, but actually point you into a great direction what you need to do. So great presentation.
Thank you. Any other questions or statements?
Great job, Elijah. I'll just say I really appreciated how you set the stage for helping us to understand how devastating a disease these gliomas, especially pediatric gliomas, are. We have not made progress in this area of cancer for years, and it is almost a universally fatal diagnosis within 2 years. So the work that you've done to be able to identify better ways to ensure that CAR T cells are able to provide efficacy will really make waves with lots of other very difficult to treat cancers. So amazing job. I'm a little biased because I do CAR T cell work, but you 100%, I think, clearly understood the rationale, understood what you were doing, the experiments, and the really important clinical implications down the line. So great job.
Thank you, Dr. Rouse.
I think I want to make a comment. I think the previous presentation gave us a background about pediatric brain tumor, how deadly it is, right, and how badly hurts the community. And Elijah's presentations point out what is the current way possibly can cure those deadly disease. So I think it's actually put them side by side, is a good introduction, and some of the pioneer study currently available. It's very good for our audience to understand. So thank you again for the wonderful wonderful presentation. Our next presenter, thank you, is Lataran McKinney.
Hello, my name is Latoria McKinney. I'm a senior here at Texas Southern University. I'm a biomedical science major. Okay, so the title of this is Alive. So it's an African American liver cancer genetic epidemiology study. Okay, so the introduction. So hepatocellular cancer is a rapidly increasing, highly fatal cancer. Major risk factors for HCC in the United States include hepatitis virus, heavy alcohol drinking, hepatitis B infection. Recent studies reported the absence of any these known major risk factors in a large population, well, proportion, sorry, 20% to 40% of patients with HCC. There is limited data on the risk of HCC in patients with non-alcoholic fatty liver disease, and the data that has been found mainly is for white American males. This rift in health knowledge creates a major health disparity for African American men and women. Okay, and then some of these disparities are provider-based. Patients with HCC who are treated at safety net hospitals are less likely to go under surgery, and those who do receive fewer liver transplantations and undergo more procedures. Surgery is also less common at community versus academic cancer centers. However, hospital variations does not fully explain the observed survival inequalities. Okay, so the hypothesis, African American men and women are more likely to develop liver cancer than the non-Hispanic white men and women. This study aims to identify both genetic variants and health disparities that contributes to that higher risk. Okay, so the methods that we did for this study included contacting potential participants via the Texas Cancer Registry, surveys, saliva kits, and mailings. So the mailings, if they wanted to do the mailings, they would mail out the surveys to them. And if they completed the surveys, then they had the option to do saliva kits, which would take their saliva samples and do studies to see if it was like a genetic factor why they had liver cancer. So it's an ongoing study since 2022, so the results are not conclusive just yet, but this is what I have found. So most of the patients who completed it were males compared to females, and it's based off 99 completed surveys. Majority of the patients were seen to answer more during the times of 12 p.m. to 2 p.m. and 4 p.m. to 6 p.m. So the conclusion, participants have been seen to answer more via calling around the times 12 p.m. to 2 p.m. as stated in the last slide. And out of 909 patients via the Texas Cancer Registry, only 99% have completed the surveys, which is a 10.9% participant range. So, but low, I've completed more results. So mainly the patients completed it by mail, and mainly the stages were tumor stage 1, which is, they called it early. So, yeah. And yeah, this is acknowledgement, so I just want to acknowledge everybody from the P20 to TSU and then my lab team.
Thank you. Is any question? I can see you're a little bit nervous. So I actually have a question for you. What do you think your study can point out a direction how the future kind of survey or anything like that can be handled according to what your research result?
Well, I was gonna say, well, when the patients did answer, most of them did not want to participate because they already said they go through a lot when they're doing treatments. So I would like to know, like, why did they not want to do it? Like, what in the process of being treated for the liver cancer that they made them not want to participate in the surveys? Awesome. Well, yeah, they answered around the time. They just didn't want to participate. Yeah.
Oh, these are the people who called when you got them on the phone, and they said no?
Not all of the patients, not all the 99% were the ones that answered. That was just the ones who participated, so that could have been over the phone or through mail.
I was curious about the ones who answered the phone. What age group do you know?
Yes, most of the participants are like in the age groups of the 60s, like 65 is the like median age. The lowest age I've seen is like 28 as the lowest patient with liver cancer.
Great job. Do you think, as you think about surveying patients who are undergoing treatment for liver cancer, which is quite extensive and often including liver transplant, do you think that perhaps approaching them during one of the long days on clinic where they're maybe in the waiting room or have some downtime, or partnering with clinicians who are taking care of the patients to facilitate completing the survey would be helpful?
I think so. I think more patient contact in person would be better instead of like on the phone or through mail, because not as personal. So they really don't see the point of doing the survey because they don't feel like it's personal. They just feel like somebody wants to know their business.
Absolutely, and perhaps partnering with the team of folks who's physically taking care of them and along the ride. Okay, awesome. Thank you. Great job.
Thank you for the second session, all the great presenters. I really, truly enjoy your presentation. Let's take another 5 minutes break. I think we're catching up. All right.
Hello? All right. All right, guys, it's time to settle down and get back. We just finished the second 5-minute break. Don't worry. This is the next round of podium presentations. We're almost there. So I have the pleasure of introducing Dr. Estes. She is a PharmD, a RPH, is an associate professor of pharmacy practice for Texas Southern University College of Pharmacy and Health Sciences and clinical faculty pharmacist specialist. Drug information at Avenue 360 Health and Wellness. Dr. Estes has over 20 years in academic pharmacy with clinical research and health disparities with a focus on genetic renal disease, specifically polycystic kidney disease, serving as PI for the Texas Southern University Research and Innovation Awards, exploring the genetic component of chronic kidney disease. The Community of Council of Greater Dallas and Texas Southern University, COPUS, All of Us Research Program Outreach Innovation Initiative, that discusses medical mistrust and vaccine hesitancy in the minority community, especially among African Americans, past and the present, and now in the area of COVID, as well as a co-investigator on numerous projects. Dr. Estes is a faculty research mentor in various universities' summer undergraduate research program, including the TSU LEAP program. Dr. Estes is the TSU co-lead of Collaborative Cancer Research Education Program, CREP, within the Baylor College of Medicine and Texas Southern University P20 Collaborative Union for Cancer Research, Education, and Disparities Program. Please welcome Dr. Estes. You're welcome. Nice to meet you.
All right, guys, I think we're off to a very good start. Everything has been wonderful up to this point, and we're going to keep it going. So our next student that we have, or scholar, is Iman Murray.
Greetings, I'm Iman Murray. I'm a first-year pharmacy student from Chicago. And today I'm going to be going over FAT1-mediated CDK4/6 inhibitor resistance in ER-positive breast cancer among African American women. So ER-positive breast cancer has estrogen receptors, and estrogen-driven activation of cyclin D CDK4/6 kinase is crucial for mammary development and proliferation. Clinical trials have confirmed that CDK4/6 inhibitors work for treating cancer, and some of those include palbo, ribo, and abema. The role of FAT1 is a tumor suppressor that helps regulate how cells move, die, and grow. And then the impact on African American women. African American women have mortality rates 40% higher than any other race when it comes to breast cancer. So the hypothesis is FAT1 gene alterations cause resistance to CDK4/6 inhibitors in ER-positive breast cancer among African American women by disrupting cell cycle regulation. The methods I used was the clonogenic assay. For the clonogenic assay, I used FAT1 parental and FAT1 ribo-resistant. And for those, we treated with CDK4/6 inhibitors and let it grow over 2 weeks and then stained it. For the other method I used was SRB assay, and I used FAT1 knockout cells and with CDK4/6 inhibitors for 72 hours, and then I did the staining after that. Okay, so these are the results. If we look at the clonogenic assay first, for the ribo-resistant and the MCF7 parental. So MCF7 is a breast cancer cell. So as you can see, for the MCF7 parental, the growth was much more than the ribo-resistant. And then also for the SRB, we use MCF7 and MCF7 FAT1 knockout. As you can see, MCF7 had lower cell survival than MCF7 FAT1 knockout. For the MCF7 Fab1 knockout, it was around 80%, and the MCF7 it was around 60%. So the conclusion is that we found that cells that were altered or had knockout FAT1 were more resistant to CDK4/6 inhibitors. The MCF7 parental cells show substantially, higher growth cells than ribo-resistant cells, indicating that the adaptations are significant. Loss of FAT1 tumor suppressors promotes resistance and increases CDK6 by disrupting the hippo signaling pathway. And so lab-based study is, we need to do further research in living organisms to confirm. And also it is being studied that CDK4/6 inhibitors are effective for ER-positive breast cancer and resistance is common. Studies are showing that upregulation of CDK6 kinase could be a key resistance mechanism. And I just want to acknowledge the P20 Cured Scholars Grant and everybody involved, and specifically my mentor, Dr. Benny, and all of my lab partners.
Don't leave. We have any questions for Iman? Questions? Dr. Rouse?
Great job, Iman. I'll point out that Iman is working on one of the pilot projects, which is really exciting. So as you were understanding and learning about the background of why this project was important, were you surprised to learn that the mortality or outcomes, particularly for Black women with breast cancer, were substantially higher or worse than for white women with the same cancers?
So I have previous knowledge with breast cancer, especially with Black women. So when I originally found it out, I was surprised, but it made sense because majority of the people I know have breast cancer, like in my family have breast cancer. So yeah, because it is significantly higher for Black women, and in general, we get it at a more aggressive rate too. That's what makes it worse.
Absolutely. And so we're often trying to figure out, is there what we call a quote unquote biological difference? Is there a difference in the way the tumor behaves? Is there a difference in how Black women respond to drugs? But then also we have to think about the other differences. So you mentioned like coming to care at a later time point and having more advanced cancer. Do you feel like doing this research project in the lab gave you a bit more insight into the fact that it could be quote unquote multifactorial, that there could be multiple reasons why Black women do worse?
Yeah, I feel like this helped a lot because you can see the differences of how the cell progresses. So that makes a difference because it helps you understand if it's an issue with the cells in general or if it's just specific person issues. So I think that was the biggest difference I've seen.
Absolutely. So you got some disease specific and perhaps patient or population specific, right? Great job, Iman.
Thank you. Good job. Okay, so we're going to move on with Brandon Odendina.
Oh, good morning. My name is Brandon Odedina. I'm a recent college graduate from Texas Southern University with a Bachelor of Science degree in biology, and I'm also a rising first-year pharmacy student. Oh, thank you. Oh, I gotta introduce the title. Thank you. So, over the summer, I worked at the Texas Children's Pavilion Tower on in Dr. Lupo's lab on a project that involves assessing the correlation between epigenetic age acceleration and the time to diagnosis for obesity and acute lymphoblastic leukemia survivors. So acute lymphoblastic leukemia is a cancer that affects up to 4,000 individuals every year in the United States. And it's a pediatric cancer, so the age range is usually at less than 15 years of age. So although acute lymphoblastic leukemia, or ALL for short, is the survivorship for the cancer has increased over the last decade, the survivors may experience increased susceptibility to comorbid health conditions. So epigenetic age is the biological age of an individual that is determined by examining the patterns of DNA methylation, and DNA methylation is the addition of methyl groups to CpG sites on DNA. Epigenetic age acceleration is when an individual's biological age is actually higher than oh, thank you. Their time-based age, I guess. I should just say that instead. And it's, yeah, when that occurs, it usually means they're also more prone to health conditions and diseases. So with this survival analysis, we sought to observe and see if there's any correlation between epigenetic age acceleration, or EAA, and the time to diagnosis for these comorbid health conditions in ALL survivors. So we hypothesized that there is indeed a positive correlation between the epigenetic age acceleration in ALL survivors and the time to diagnosis for these health conditions. So this survival analysis used a diverse cohort of 68 participants, and inside this cohort, there was a large number of Hispanic individuals. Additionally, the male to female ratio was about what you would expect from a study on ALL survivors, because there was a larger male to female. There was more male than more male than female diagnosis. And also, there were other clinical and demographic factors we tested for, but there was no statistical significance except for the Yost index, which is an index used to measure social determinants of health. The participants included individuals diagnosed with ALL at Texas Children's Hospital from 1992 to 2013, and they were seen at least once in the long-term survival clinic. Germline blood samples were collected at first clinic visit, and demographic information was extracted from the EHR. So the methylation array data was pre-processed and controlled, and the methylation data was also processed was also processed using MinFeed, which is a package in the bioconductor computer software. And also the EAA was estimated with the Horvath calculator. Finally, the relationship between EAA and TEM2 obesity and neurocognitive ADHD and learning disorders were almost all determined by Cox regression, with the survival package in RStudio. So here we have a Kaplan-Meier plot, and the Kaplan-Meier plot is usually used to show the relationship between a factor of interest and the time to an event. In this case, that event is the diagnosis of obesity. And the x-axis, here, we have the years since the participants were diagnosed with ALL, and the y-axis, we have the survival probability, which can also be seen as the proportion of individuals that were not diagnosed with obesity. So in the first quantile is the participant with the lowest amount of EAA, and in the fourth quantile is the participants that have the highest amount of EAA. As you can see in the line plot, or the KM plot, the first quantile has a proportion of individuals that experience the least amount of diagnosis for obesity, and the fourth quantile has the highest amount, the highest proportion of individuals that experience the diagnosis. We also, with the Cox regression, we found that there was a hazard ratio of 1.13, a p-value of 0.009, and a 95% confidence interval of 1.03 to 1.2, which shows statistical significance. So in conclusion, we found that there is indeed a positive correlation between EAA and time to diagnosis for some comorbid health conditions. In this case, that was just obesity. We didn't find any statistical significance for the neurocognitive ADHD and learning disorder diagnosis. And I just said that. And so this study it strengthens our understanding of the role that EAA plays in the susceptibility to CHCs in long-term cancer survivors, or ALL survivers. So, first of all, I want to thank the P20 Cured program for allowing me to learn a lot this summer, and also my mentors, Dr. Shure and Dr. Lupo for introducing me to this topic, and also Christina and also Christina for being there for me the entire project, and also Dr. Richard for doing all of the EAA calculations as well. And yeah, that's my project. Thank you.
Any questions? Great job.
Amazing job, Brandon.
Yes.
I was just texting with your mentor, Dr. Schurer, and he was on Zoom and had to get off, but this is a quote, Brandon killed it. He did amazing and said it was a joy to work with you. So we are so very proud of you. As a pediatric ALL physician who takes care of patients who are at the highest risk of relapse, this work is so important. We're still constantly trying to understand the outcomes of some patients, especially patients who are underrepresented, and why they are worse than other patients. So we are so very proud of you. I think you did a great job. And I would ask, I guess, what was the most surprising thing that you learned during the course of your research?
Well, I think my most favorite subject for sure was definitely the epigenetic age concept and everything. Yeah.
Who knew we have different ages,
yeah,
right?
Exactly. Yeah, it's really interesting. I found it really interesting as a topic.
Absolutely. Thank you, Brandon.
Thank you.
Hey, Brandon. Brandon, I just want to say, great job.
Thank
and
you.
Such complex presentation, and you made through so beautifully, so smoothly, and so proud of you. Congratulations. And also, I did not know before your talk, I did not know that obesity and ALL is correlated. So that's great.
Oh, yeah.
A great scientific information, and thank you. Great job.
Thank you.
Any other questions? All right, great job.
Can I really quickly, Dr. Estes, I just wanted to introduce one of our Baylor College of Medicine leaders, Dr. LaChauncey Woodard. Stand up. I'm going to embarrass her. Dr. LaChauncey Woodard is our fearless leader of the Office of Community Engagement and Health Equity at Baylor College of Medicine. All of y'all met her at orientation. She facilitated some really cool games. And also really, I think, wholeheartedly welcomed y'all, and the office also sponsored some of our lunches. So we're so excited and honored to have the full support of Dr. Woodard, who ran over here in between doing a bunch of things. So I just had to introduce her before she has to get back to patient care.
Awesome. Thank you.
Hey guys, so it's so great to see you all again. Some of you all reached out about some mentoring meetings, if that happened, and those haven't been scheduled, which I really think they haven't. Please reach out to me again. As I told you guys, my email is lwoodard@bcm.edu, pretty easy. Dr. Rain has it Dr. Rouse, okay, I have a Dr. Rain that I work, has it. And also, I just wanted to say, congratulations on the amazing work that you all have done this summer. It is so important that we have young scientists like you all who are coming behind and learning these skills and blazing the path and helping to create more equitable healthcare and research for all of us. And you guys are going to be the ones who are making the scientific discoveries, and the doctors and nurses and health professionals who are taking care of me soon. So pharmacists, all the things. I did say health professionals because I want to make sure I catch everybody. So make sure that you guys keep doing the great work that you're doing. We're really proud of you, and I'm so excited for the small role that our office has in supporting the great work that's happening here, and love the opportunity that we've had to collaborate with TSU. So thank you guys so much for all that you did and giving your all this summer. We appreciate it.
Thank you, Dr. Woodard. So our next scholar is Angel Reed.
Good morning. My name is Angel Reed, and I'm a sophomore at Texas Southern University. This summer, I had the pleasure of working with Dr. Kahali, and our experiment was exploring intellectual outcomes after proton radiotherapy or surgery only for pediatric brain tumors. So the present study examined neurocognitive change and functional outcomes in pediatric brain tumors treated pediatric brain tumor patients treated with proton radiotherapy. Both radiotherapy and neurosurgical resection are associated with neurocognitive risk. This is an ongoing study, so there's no consensus in the field, but there is literature that states there is greater neurocognitive decline associated with surgery rather than proton radiotherapy. The impact of radiotherapy and neurosurgery on psychosocial outcomes has not been further explored, which is the purpose of our experiment. Better understanding of neurocognitive outcomes in this patient population will help us to identify children at greatest risk, and it gives new hope for future pediatric patients with brain tumors. The purpose of the work conducted in Dr. Kahali's lab is to improve cognitive and psychosocial and the quality of life in children who are treated for brain tumors. Part of that is evaluating the neurocognitive and psychosocial trajectories of this population. So it is hypothesized that children who experience greater neurocognitive decline after treatment will experience worse psychosocial and behavioral outcomes. So the methods that we used is that we enrolled 134 patients between the ages of 3 and 18 at the time of radiotherapy or neurosurgery. The patients had either only radiotherapy, radiation, or surgery. They didn't have both. Patients came in for annual cognitive testing, and some of the tests that we used were DCAFs. DCAFs focuses on executive control. We also gave briefs, which is the perspective of executive control. We gave them both to parents and the children patients. And we also gave WISC, which is another type of IQ test, and it tests like mobility and if children can switch between letters and numbers. We also looked at the changes in IQ between baseline and 2 years post-treatment, and we administered surveys to both children and parents to determine interpersonal and social factors. So the results on this table, we can see that we have over a quarter of our patients experience 5 or more points in IQ decline between baseline and time 3. And the bottom table shows that about 40% of our patients had surgery only, and about 60% of our patients had radiotherapy. This WISC normal curve shows the scores between IQ, and this is how we scored our patients after IQ tests. To conclude, the limitations of this study included sample size, because we only were able to enroll 134 patients. Many pediatric brain tumor patients who experienced neurocognitive decline followed treatment. Greater neurocognitive decline was associated with worse behavioral outcomes as reported by parents through the surveys. Differences in parents' and patients' perceptions of behavioral functioning were identified, and this study will eventually help us to identify children at greatest risk for functional difficulties and survivorship. I would like to acknowledge everyone in the P20 Cured program and for allowing me this great experience. And I also want to acknowledge my lab mentor, Dr. Kahali, and the Kahali lab team. Thank you.
Any questions? Questions, comments? Dr. Rouse?
Anyone else? Okay. This is great job. This is such an important topic. And just to level set, when we diagnose a child with brain cancer and radiation therapy is a part of the curative plan, and we tell them, unless we do radiation therapy, your child may not survive, it's a very difficult position for a family to be in. And one thing that we, as you said, we don't quite understand the longer term cognitive effects. We think we understand, but we certainly don't have them laid out. So this was such an important project that you did. And I wanted to ask if between you and Dr. Kahali and the research team, you've thought about how this data will be presented perhaps to families when they are coming in to hear about treatment that includes radiation, and if you've identified any potential ways that we can help prepare them for these potential outcomes, including decrease in IQ and what that means in the real world for them.
So I think that's like partially what the survey is for, because on the survey that we give to the parents and the students, we ask the parents, okay, like how are they behaving socially? Do they have a problem making friends at school? Do they behave at home when you tell them to do something? And usually, like when we look at the difference in the surveys, we see that the child thinks that they're fine, but the parent is like, okay, the child is not fine. So the children don't really understand, you know, the severity of the situation. But I think through the annual cognitive testing, since it's only one time a year and since testing is so long, it kind of helps the parent to be like, okay, like this is what's going on, and we can help the teachers to understand as well.
Great job.
Great talk. So for other types of neurocognitive disorder, like cerebral palsy, like they found that early intervention is like really key in helping the child to, you know, reach their potential or progress. And I wonder if you know if anyone is working on like early intervention either before radiotherapy or shortly after, if that can help these outcomes.
I'm actually not sure, but I can check in with my mentor and get back to you on that.
How you doing? And thank you for having me. As a psychologist, I am actually interested in were there any negative patterns of cognitive decline? So when you guys actually had the baseline for this population, either radiotherapy or neurotherapy, did you see when you say psychosocial, what kind of decline does that include, say psychosocial?
So there's decline in the mobility. So usually when we give tests, we test them, can you build blocks? If the color says red, but it's in blue ink, will you say blue or will you say red? If I give you a grocery list, can you tell me can you repeat what I said back to you? Do you know what everyday words mean? There's vocabulary test. Yeah, there's just different type of tests. And since it's annual, it's one time a year during testing. Usually when we see kids, it's like, okay, this is your score. And when they come back the next year, there's something that happened between that year where they come back and they do the annual testing again. And it's like, okay, you're not performing as well as you were a year ago.
Any other questions? Great job. Thank you. Okay, and our last scholar is Amaya Tootle. Are y'all clapping because it's close to lunch or what?
Absolutely. Thank you, ma'am. I understand it, but bear with me, please. I know we're all hungry. My name is Amaya. I'm a senior here at Texas Southern studying biomedical sciences in the College of Pharmacy and Health Sciences. My project is Guardians of Immunity, evaluating the frequencies of immune cells in subtypes in CD101 knockout and CD101 wild-type mice. I was working in the Hudson lab under Dr. William Hudson with my mentors, Colby and Sean. So our CD8 T cells play a vital role in our immune defense, most notable in killing cancer and virally infected cells. Cancer cells inhibit CD8 T cells via inhibitory receptors on the cell's surface. Studies show that we can reinvigorate CD8 T cells in cancer by blocking these inhibitory receptors. CD101, as we can see in figure 1, the purple, is a surface protein implicated as an inhibitory receptor on T cells, but its exact function is unknown. However, what we do know is that when upregulated, CD101 is upregulated in dysfunction of CD8 T cells. So we can see in figure 2, this represents CD101 expression in CD8 T cells. When there is 0 expression of CD101, that indicates that a cell is naive or it has not encountered its antigen yet. And then once we do start seeing CD101 expression, we start to see the dysfunction of a CD8 T cell, meaning that it is continually encountering its antigen, but not efficiently or effectively killing its target cell. So what we want to know is which immune cell subtypes express CD101 in CD101 wild-type mice, and then we want to know the frequencies or their phenotypes of any immune cell subtypes altered in CD101 knockout mice relative to the wild-type control. The goal is to determine which immune cell populations express CD101 in the spleen, thymus, and bone marrow of CD101 wild-type mice. And then we'll compare those immune cell frequencies between CD101 knockout and wild-type mice. So this is a multi-step method. We use a mouse model. First, I isolate the tissues. So I harvested the spleen, the thymus, and the femur for the bone marrow to disassociate them into single-cell suspension by manual tissue processing and centrifugation. Next, we created a 30-color antibody panel to label each sample. Label each sample. In step 2. In step 3, with that antibody panel, we first counted them using flow cytometry, and then enumerated each cell type using the 5 lasers within the flow cytometer. So as each cell passes through, a laser hits its marker, which is our antibody that we labeled earlier, and it helps, it glows its respective color, which we analyze in step 4. So in figure 3, we identified 19 major immune cell populations, and we're able to compartmentalize them into 5 groups. So this is myeloid cells, CD4 T cells, CD8 T cells, CD8 positive T cells, CD4 positive T cells, and innate lymphoid cells, as well as B cells. In figure 3, we see that in CD101 wild-type mice, CD101 is expressed in 5 major subpopulations, including neutrophils, type 2 conventional dendritic cells, macrophage, CD8 positive effector memory cells, as well as CD4 positive Tregs. In figure 5, we see that there is no standing observed in these populations, meaning that confirming genetic deletion or global genetic deletion of CD101 and the specificity of our staining. In figure 6, we see that the immune frequencies are pretty similar between wild type and knockout mice. And then figures 7 and 8, we see that there were trends in the population where CD101 was detected in wild type, and then we see that there were differences in frequencies between CD101 wild type and knockout mice compared to CD101 wild type, where there were increased frequencies of CD8 effector memory cells, Tregs, and macrophage, as well as decreased frequencies in neutrophils. So in conclusion, we developed a 30-color antibody panel, which is the first of its kind. And this allowed for us to do a detailed simultaneous analysis of multiple immune cell populations and their subsets. With this, we were able to identify 19 major cell populations and determine which populations express CD101. CD101 wild type and knockout mice do differ in frequencies of populations that express CD101, suggesting context-dependent regulatory roles of CD101. and the global effects of CD101 deletion should be considered in future translational research. So some limitations of our project was that we used a small sample size in MECOS 3, so we used 3 wild-type mice and 3 knockout mice. And due to the expression of CD101 on multiple immune cells, population CD8 positive T cells, T cell targeted models should be used in future studies for CD8 positive T cells to better understand how CD101 functions intrinsically. Overall, this study shows that CD101 is important for the immune system's development, and this finding does support the idea of targeted CD101 immunotherapy. A specific one is called checkpoint blockade, which helps the immune system detect and destroy cancer cells more efficiently. I would like to take the time to acknowledge the Hudson Lab. Actually, my mentors are here right now. Thank you for your patience. I appreciate it so much. As well as the P20 Cured Scholars Grant and the Cancer Research Education Program. The honorable mention is Ms. Nikisa Saab, who is not here today, but if it weren't for her, I actually wouldn't be here. Something that messed up with my application, I didn't know. I went to her office, and within the week, I was here. So I really appreciate her. Questions? Oh my gosh, that was so fast. Okay.
A lot of work, Amaya. Wonderful. Since your PI is here?
My mentors are here.
Your mentors are here? Cool.
Okay.
So my question is sometimes you get an opportunity to publish your work. And since this is technology development, I'm assuming?
So the panel is the new development. So we used, based on past projects done by the Hudson Lab, as well as an OMIT paper, we were able to handpick a panel that would best fit for what we're trying to do, which is determine the multitude of immune subset cell expression. So I learned I had to add that we developed this panel because they emphasized to me that this is unique. This is the first of its kind, and it will be used in future projects done by their lab. I don't know about others, but yes, I had to add that.
So what's the opportunity to continue the work?
To test it, so they actually let me know that they are running it on panels that they're doing in these coming weeks to see how effective the expression is in their samples. I guess the opportunities are kind of limitless with this. You can use it for, I guess, multiple projects.
For you to continue the work.
For me to continue. Yeah. That's between me and them, I guess.
Hopefully we'll have some future
I'm putting it out there.
for you.
Okay.
Hey, Maria, that's great work.
Thank you.
And so much data over there, and then you present really well the graphs for the illustration about the flow. You made a complex technology so simple. Great job.
I kept asking them, like, explain it to me like a fourth grader. Explain it to me real slow.
Yeah. I remember before the program, before the technology week, both you and your mentors asked to be trained in this core lab of local. Has the technology week's training helped you in this project?
I'll be so honest, the technology week flow was harder than what I learned with them.
Right.
But when I got up there, I was able to just kind of hop on. Like I had a better understanding of how everything was working.
That's fantastic. It looks like next year our training needs to be a little...
I don't think it's that. I think his name is Joel. He did a really great job. He loves his job. I will say that. It's just information overload. There's a lot going on up here.
All right. Sounds great. Congratulations on great job. Well done.
Thank you.
So shout out to the advanced technology course that Dr. Shisha helps lead, and Joel Cedarstrom, who in Flow, who is amazing and taught me how to do Flow. I just want to congratulate y'all, because if y'all have never done flow cytometry, to create a 30 color panel.
Yeah, it's a lot.
Is a great feat. So mentors, go up there and we'll take a picture. Thank you.
I also have a comments, Amaya. I think when I kind of go through the presentation with you, you also mentioned animal study you have been doing, right?
Yes.
That was really impressive. She said she never.
Oh yeah, I've never touched a
done
mouse
any
before.
animal before.
Never touched one. Never considered touching one. And then they really just.
Yeah, so that was quite impressive. Thank you, Dr. Hudson and the lab. I mean, your lab is amazing. Yeah.
As they come up. Which one? Right here. Right here. So that concludes our oral presentations, and I think we need to give a round of applause to all of our scholars for doing such an excellent job. For many of them, this was their first time actually doing an oral presentation, and I know that they kind of struggled with trying to put it together, their abstracts as well, and I think they did an outstanding and marvelous job today. So congratulations to all of you, and good job. So I'll go. Lunch break? Lunch break. Okay, lunch break. Hold up, hold up. Oh, instructions forthcoming.
All right, guys, great job. It is now time to eat. You have a question? No, you don't get a mic. So we are going to pick back up at 1, and I am actually going to do by tables, so that way it keeps us organized and it doesn't overcrowd Sodexo. So what we will do, yes, okay, come on y'all, go ahead. Y'all are hungry, go ahead. Y'all are hungry. And also, do not forget to scan your survey and do your survey. Oh, well, you have now gotten the instructions. Y'all cannot leave until the surveys are done. Okay, scholars. I hope you all have enjoyed lunch. We are getting ready for the poster session now. Come on now. Where's the clicker? Where's the clicker? Where did the clicker go? Yes, we're gonna make sure that your parents can see on Zoom. So I guess I'll do my own introduction. Okay.
Beyonce don't introduce herself, so why would Charmise? Okay. Ms. Charmise Buck is currently a pharmaceutical sciences doctoral student, whoop whoop, at Texas Southern University. Ms. Buck has 2 masters, not 1 but 2, okay, in forensic science and crime. Wait, you a CSI? I didn't even know that. From Stevenson University, and her bachelor's in chemistry from Hampton University, another HBCU. Whoop whoop. Ms. Buck is the CEO, wait a minute, of Shalom Analytics and Consulting, LLC. Shalom is an analytical consulting laboratory, and over the past 10 years, not only has Ms. Buck furthered her education, she has worked in the clinical toxicology field, cannabis, analytical chemistry industry, did bioterrorism research for the Homeland Security and FDA. Whew. In her spare time, which clearly she has very little of, she enjoys building her business, reading, serving the Lord, teaching at her church, and giving back to her community. Ms. Buck is also passionate about mentoring and teaching youth about STEM, and I cannot say how excited and grateful we are to Ms. Charmise Buck for all of her contributions to our program, and we welcome you.
Thank you, Dr. Rain, for that beautiful introduction. It is a blessing to be here. I am so, so proud of y'all. Even just coming on at the end of June until now, like, I've taken you all under my wing. Y'all aren't going anywhere. But we are getting ready to get into the poster presentations. We have 5 incoming freshmen that did research this summer, and they are going to present. I am so proud. So I'm actually going to introduce each freshman, and then you can go stand by your poster, and it's just going to be rounds of people just walking up, listening to you, present about your research, and remember, if you do not know the question, say, I do not have the answer for you at this time, and I can get back to you. Never give an incorrect answer. It's okay to not know the answer, okay? All right, so first we have Mr. Corey Cox. That's right. Corey is an incoming college freshman at Texas Southern University, planning to major in biomedical sciences. Upon graduating college, his ultimate career goal is to continue working in the field of research. Mr. Corey, could you please, well, you already beat me to it. All right. Next, we have Ms. Gabrielle Graves. She's an incoming freshman at Texas Southern University and planning to major in biomedical sciences, as she walks over to her poster. Next, we have Ms. Zion Abira. She is an incoming freshman as well, and she's majoring in clinical laboratory sciences. Her current interest is a career in medical laboratory science with emphasis in hematology oncology. Go, Zion! Next, we have Miss Miracle. I'm not going to butcher your last name. Miracle is a rising freshman at Texas Southern with aspirations to attend pharmacy school. Go ahead, Miss Miracle. Next, we have Miss Soraya. Soraya Scott is an incoming freshman biology major to Texas Southern University. Prior to TSU, she was a member of the Technical National Honor Society. She is committed to reflecting her dedication and determination to pursue a career in the medical field. So now the poster sessions have begun. Y'all can go ahead and walk over to the posters. All right, everyone, we have 3 minutes left. Well, really, 2. We have 2 minutes left, so let's begin to wrap it up on the poster sessions. You all did a phenomenal job in explaining your research and answering the questions. So just leave the posters pinned up, please. But we're going to wrap it up soon and continue the next part of the program, which we will move into the presentation and awards. So, all right. I'm going to go ahead and hit the next. I am going to introduce Dr. Shisha. I love Dr. Shisha. So Dr. Shisha is a professor in the Department of Molecular and Cellular Biology of Huffington Department of Education, Innovation and Technology, Director of Antibody-Based Proteomics Core at Baylor College of Medicine. With expertise in cancer research and advanced technology development, she has served as core director at the Genomics and Proteomics facilities, developed advanced technologies in genetic engineering, genomics, and proteomics, supported research programs, and co-authored over 80 publications. Wow. Dr. Hung provides advanced technology, education, and training to students and faculty at academic institutions and secondary school communities, including establishing and leading biotechnology incubator for teachers and students with the support of NIH R25 Science Education Partnership Award. Dr. Hung is the BCM lead of collaborative cancer research education program with the BCM TSU-MH P20 Collaborative Union for Cancer Research Education and Disparities Program. Please welcome Dr. Shisha.
All right, thank you so much. Thank you, everyone. This is the best part of the day or of the summer, right? Congratulations to every one of you, the scholars. So that's his first quiz. It's about the cured program. Who can say it fast? Okay, not a real quiz, but you will know already or after this. So again, congratulations. So we are going to hand out the prize for everyone. So all of you for the presentations, great job. I learned a great deal of the research in different areas, breast cancer and AML, ALL, all kinds of areas. Thank you so much for doing such a great job at the oral presentation and also at the posters. So a lot of things are new to me, and you explained so well at the posters and also oral presentation. It's very impressive. So as I talk with Zion, I did not do a presentation like this until I was in graduate school. So you really are doing, have such great opportunity of doing so well. So now it's the prize time, and all the CREP leaders, please come. Deborah, Juan, and Flora, we're going to give the awards to everyone. And then all the scholars, please come up. We're going to give award to every of you, and then we're going to have a big happy. Oh yes, we are not going to keep the bags, so we're going to give the bags to you as well. And should we call one by one or just have everyone and then?
Let's do one by one. Yeah.
All right, call one by one.
Where are they?
Nabrios.
Congratulations. Thank you for coming.
Yeah, so we need you to stay over here for picture. Nora. Taylor next. Devira. And Ara. All right.
And you get
your
it
next. Alexina. Yes.
Oh, yes.
Corey. And Taylor Forwell next. Gabriel. Congratulations. And Zion. Chinaza. Chizara, right?
Yes.
Wonderful, thank you.
Elijah.
Elijah. Lotorian. Lotorian. Ima. And a miracle.
Here you go. Miracle.
Oh, the bag. The bag. Brandon. Yeah. Let's go out to the front. All right, congratulations. Angel. Saraya. Oh, of two. Sorry, and Amaya. This is yours, right? Amaya. Congratulations, everyone. You have done such an amazing job. So now we're going to have a big group picture. And how are we going to take the group picture? And also we need the leaders come and the Rain, Veronica, and all of us, yes, in the group picture, the PIs as well, the mentors, please come. We're going to have a big happy family picture. You all look amazing.
And, okay, she will tell us.
I'll go this side. Yes, the mentors. Do we have any more mentors? Come into the pictures as well.
Let's do one with just the scholars and then one with everybody who is here to support. So just scholars included in and all the. Never see them.
Okay, everyone, congratulations. I think the last item is to make sure what do we need? The survey. We need to have the survey done today. And also, I still need yes, QR code on the top. Yeah, so for Baylor College of Medicine badges, you can either turn in now to Dr. Ross, or you can come in tomorrow, Friday, to turn in.
Or last resort, Monday. Okay?
Yeah.
So that's the official last day. And I already talked to y'all about your supplemental stipends. Okay, who has a badge? I have no supplemental. Thank you. Y'all have a little bag? We can get something to put it in. Okay, cool.
So please scan the QR code. It's right on the screen, or it's in your packet. The agenda also has the QR code. We value your feedback for this past 8 weeks. This is the first of many P20 cured cohorts to come, so your feedback is very much needed and appreciated, please. Yes. You can't leave, says Dr. Murray, until you complete your survey, please. So take out your phone, and thank you for working on that. While you guys are doing that, just a few additional announcements. Like we all know, the 8-week session has come to a close, but the year-round commitment continues. So with the year-round commitment, you guys are expected to participate in minimum of 3 community outreach events. During the past 8 weeks, you were required to do 2 community outreach events. Several of you did, but some did not. So for those that did not complete their community outreach events, Dr. Estes has actively tracked your records. Yeah. So you will still be required to do those 2 events. So if you did 2 events over the past 8 weeks, then you have 3 more to complete. Otherwise, you have a total of 5. Dr. Estes will be sending you additional information. And we have listed on the slide priority events. 1 in September, 1 in October, January 2025, April 2025. So we need to keep the conversation going. Our doors are open for continuing mentorship, guidance, and career development. I would... Yes, thank you. Dr. Estes has a few more information about the outreach events.
So as part of community outreach and engagement, there is one last assignment that you guys have to do. And that is a reflection on your involvement in your community outreach activities. Even though some of you did not participate in any, the components would be for you to reflect, tell us about how your community activities were, and also the other would be, what place does outreach have in research? So even though you may not have attended any of the activities to date, you can still, in your reflection, address that question, okay? So we are, we'll be sending out an email that has the information, instructions for the assignment that you need to submit by 12 noon on Monday. Since you don't have lab tomorrow or on Monday, you have sufficient enough time to prepare your reflection and upload it. Ms. Buck is going to send out the email to you with the upload link for you to upload your reflection. Are there any questions? So please note that you are required to participate in your outreach, your community outreach and engagement activities. Okay, that's part of the program as well. And just FYI, for those of, for the pharmacy students, you know you're required to do co-curricular and service learning. So approval has been given by Dr. Davila in experiential that those activities will count toward your service learning and co-curricular hours that you need in the PharmD program, okay? So if you need some extra incentive, just know that you're doing double duty or double credit for your involvement, okay? Thank you.
Thank you, Dr. Estes. Thank you, thank you, thank you. So these flyers, some of them were out in the lobby when you were coming in, but again, telling you guys to not just participate. You know, this program is all about cancers, and prostate cancer remains, unfortunately, the leading cause of cancer-related death in men, and breast cancer remains the leading cause of cancer-related death in women. So September is Prostate Cancer Awareness Month. October is Breast Cancer Awareness Month. So this is your opportunity to pay it forward, to participate, spread the word, tell, you know, family, friends, you know, people all around, not just for you to come volunteer, but also be an agent of raising awareness about these two diseases. Who is excited for 2025 cohort? Who is ready to be our official ambassadors, right? So save the date, more information to come. Each of you 19 Cured scholars that are wrapping up today will now step into the role of becoming ambassadors for us, so that way we can continue to recruit. Yes, Dr. Ross. Mm-hmm. Yeah. Okay, good. Okay, not a problem. 1 month, okay, no problem, yeah. So, but anyway, more information will come about recruitment, which would begin in the fall 2024 for the students or scholars that we will be accepting into the program for 2025. And before I go over the thank yous, one of the scholars said they had 1 or 2 things to share with us. Alexina? Okay.
Well, before we close out, we as the cohort of 2024 want to extend a big gratitude and appreciation for the whole team and the directors as the students for giving us this opportunity to go and be immersed in this huge learning program and university that is Baylor College of Medicine, and believing in us to give us this task to go learn. And right now, we hope that we will make you proud as ambassadors and as a brand and a product of this program. So from all of us, from the cohort of 2024, really want to say a big thank you to everybody in this team. Thank you.
Thank you, Cured Scholars. Very, very much received, and we look forward to greatness, because that is, you guys have no choice but to be great in all that you do. Thank you. Shisha, you have something to say?
Yeah, so I just sent an email to every one of you for 2 things. One is for high school students. We have the amazing posters over there. I have a high school student symposium on August 10th, next Saturday. And well, our official registration is closed, but I'm opening it for all of you. We're assembling the brochure book just like this tomorrow. So if you would like to come next Saturday, please send back your abstract and your bio and picture you just already made over here. And the next week, you only need to bring a poster over there. So what the benefit of that program, besides you have your amazing resume for the Cured Scholar Achievement Symposium, you have another items, if besides the opportunity, another item to attend a high school student symposium on your resume. That could help you with your career for that. So second one is I sent you an email about, I know I sent you many emails, and thanks to 12 out of 19 who already did the Technology Week survey, and your survey helped me a lot. I read it through, I know our technical survey Technology Week needs to be the second week, not the third week. I got that next year. Rest assured, it's going to be the second week of the program. So your feedback is so important. And also today, I learned that it was too high level. So I'm going to tell all the core directors says, okay, we need to tune down a little bit, and they have more background introduction. So all the feedback is going to be really important. Please, the rest of the 7 students, I know you are busy, and send back in. I'm not going to ask you to stay over here to do that. When you go back home, look at the email and send that in. Okay, Deborah says, do right now. But I'm not going to look. It's okay, you do not do it. All right, so congratulations again. I'm going to see you, all of us are going to see you a lot and follow you to know you are going to achieve big success. Thank you.
Awesome. Thank you. And thank you to everyone supporting all the events, the staff, volunteers, interns, everybody. And I would say the 10-minute begins now. So please complete your survey. Don't leave until your survey is completed. Does everyone have the link? Oh, I see you guys standing up. Have you completed? Okay. It's in the link tree. I think it's like the third link. Okay, thank you guys.
Hello again. My name is Veronica Ajawale. I want to say a big thank you to each and every one of you for joining us for today's program. I am very certain that you have received a lot of great information that you will put to practice as we all grow together to become a healthier community. Thank you to our speaker and our panelists. Thank you for sharing today's content with us. Thank you to our locations. Thank you to our interns, our volunteers, to our sponsors and our funders. Thank you also to the staff of our program, the faculty, and the leadership of Texas Southern University for supporting us in making today's program a success. Most importantly, I want to say thank you to each and every of you, the community members. We appreciate your support. We do not want today's program to end just with today's event. We want you to continue to collaborate with us and engage with us. Our social media platforms are located at CECTX Southern U. On those platforms, you will find engaging information from today's program and all the other programs that we have going on in the Community Engagement Corps. So we implore you to continue to stay connected with us, engage with us as we move forward together in building a healthy and a wealthy community. Thank you for joining us, and we look forward to connecting soon. Bye.